uploading. Yeah, we are on live stream now. Good evening, good morning, and good afternoon to all. A very warm welcome to this joint webinar organized by AMR Declaration Trust and the Alliance for Prudent Use of Antibiotic, APUA. AMR Declaration Trust is a non-profit organization dedicated to tackling antimicrobial resistance. Over the years, the Trust has organized several scientific discussions, public education programs, expert collaborations to strengthen stewardship and infection control in India. Today's webinar is part of the World Antibiotic Awareness Week activities. APUA, the Alliance for Prudent Use of Antibiotics, was founded in 1981 by the legendary Professor Dr. Stuart B. Levy, the legendary microbiologist and physician from Tufts University, Boston, who was among the first in the world to warn about antibiotic resistance. After his demise in 2019, APUA became part of the International Society of Antimicrobial Chemotherapy, the ISAC, so that his pioneering vision can continue through global network. And today's meeting brings together AMR Declaration Trust and APUA to explore a unique theme that is crazy enough to work, wild ideas outsmart AMR. Many ideas that are mainstream today, the stewardship, the surveillance, One Health, antibiotic regulation, they were all considered crazy in early years. AMR discussions evolved from microbiology to clinical medicine, then the public health, and today it is socio-economic and political domains. To move forward, we need more bold, unconventional thinking. And this webinar is free for all the participants, made possible by unrestricted educational grants from Pfizer and Viatris. Let us begin the session. First, we have Dr. Manish S. Paul. He's going to talk about poop can also fight AMR. And Manish is the CEO of Microbiome and co-inventor of the molecule antibiotic cefepime and metazobactam, a promising broad-spectrum antibiotic that has gained global attention. We are proud to have Dr. Manish with us today. Dr. Manish, over to you.
Thank you, Dr. Ghafoor. All right, so we begin to think, we used to always say, oh shit, but we don't have to be scared of saying that word anymore because poop can also address antimicrobial resistance. So I'm going to talk about a lot of shit today, about shit and how that is such an important tool in addressing antimicrobial resistance. It's not like a word that I'm using as a curse word, but using this as a microbiologist, how important this particular tool, a conglomeration of microbes that are going to help us in addressing antimicrobial resistance. So we all are aware of one of the important parts, that is AMR. I am not going to go too deep into it because we are in the week of Antimicrobial Resistance Awareness Week, and every one of us who are here are already aware of the significance of it. But some of the tools that we need to have in addressing AMR are to reduce antibiotic use, develop new treatments, improve infection control. And developing new treatments is where a lot of push-pull incentives have come in and they're trying to do the best, but I will focus on a novel way of addressing the AMR. So some of the terms I'd like to clarify to the audience. The differences between microbe, microbiota, and everything. Microbe is a microscopic organism, could be bacteria, virus, fungi, parasites. And microbiota is a collection of microbes, which is what are present on us, in us, and around us. And gut microbiota is specifically in the gut. A gut microbiota or gut microflora are interchangeably used. The microbiome, whereas, it's a microbiome. It's a microbes as well as their genetic material combined together is known as microbiome. And the foods that the microbes use is known as prebiotics, and the microbes which are used for therapeutic purpose are known as probiotics. Therapeutic or supplemental purposes are known as probiotics. Postbiotics are the inanimate, means the dead organisms that are present along with multiple components within them which have been processed, right? These are the four things. Now, antimicrobials. We have so many therapeutic areas which we are so advancing, investing so much money in oncology and orthopedics or metabolic disorders. But I'm telling you, If we did not have antibiotics, none of these therapeutic areas can survive, because once mild infection with a resistant organism, then boom. So antibiotics form the foundation of all therapeutic areas to be successful. And this is a cartoon which was developed by Otakars from Uppsala, who started the REACT initiative. And among antimicrobials, for clarity, we have got antibiotics or antibacterials, which work only against bacteria, and antivirals against viruses, and antiparasitic against parasites, and antifungals against fungi. So coming back to the story of in the beginning, God created heaven and earth. Of course, there was also infection. We all evolved from microbes because microbes are billions of years old. And then during the evolutionary process, we are in the now, but they are since then, right? Humans are the last end of the spectrum is what we see. Because looking at this Eugenio tree of life, what we see here is bacteria started way back in their evolution, and we are just evolving, just beginning to evolve as human beings in the history of Earth and life itself. So we have to remember that how much ever we know, bacteria know much more in the genetic material. And then what happened for the diversity, there has been constant migration of people. In this beautiful science paper, what we see is people who came to India from various regions over the years. And all these things have contributed towards building up our diversity in the microbiota. So we have genes from the east to the west because they've all been incorporated into us through microbiota. Why I say that? Because humans have moved around from generations to generations. And Svante got the Nobel Prize for his the description of Denis ovans, which are the one more branch which you have identified. We do not know how many more exist, of course, but at least we know we are also evolving just like bacteria and branching out. But each of the human cells are made up of plenty of varieties of human cells. And all these cells have got quite larger cells, whereas microbes are such tiny, which you can't even see in this slide. Bacteria, here you can see the bacteria almost invisible, because microbes constitute nearly 100 trillion cells in the body compared to about 30 trillion in the human cells. So we are pretty much made out of mostly microbes. So the earlier, or the hypothesis I can say, which says even the mitochondria was a protobacterium, right? Alpha protobacteria, which lost its genetic material and become incorporated and remains as a parasite, as a symbiotic relation rather with host cell. And we are pretty much more of microbes. So microbes could be the source in which we could have a solution to address antimicrobial resistance. So we have microbes in and around us. Now jumping into this topic, our gut role in resistance. We have got gut microbiota. The GI tract is a major reservoir of antibiotic resistance genes, and there are genetic material also in the gut microbiota. Now, multi-drug resistance organisms colonization precedes invasive infections at various sites in the body. Now, all these three things put together is when infection comes into play. So, fecal microbiota transplantation is a novel intervention. And a company called as Rebiota got an approval for it recently, a couple of years ago, and US FDA has approved it as an intervention in addressing antimicrobial, in addressing, as an intervention, they've approved it. So, which is very unusual because it's such a complex mixture, right? You got poop of a person, which is partially purified and used as a transplant, and it could have diverse things in that one, but still it has been approved as a intervention by US FDA. So now we can go for FMTs. Now, antibiotic resistant drugs are colonizing in the gut. And when we transfer healthy donor feces, which is partially purified, and induce it into the recipient's gut, then we actually cause decolonization of these resistant strains. And how does it work? One is, first, there is an imbalance. So the gut microbiota is in a dysbiotic stage, that is disrupted gut microflora. Then we have a FMT cycle introducing donor microbiota that increases the diversity and microbial richness of varieties of organisms of the species is what is going to increase. And then there is a dominance of beneficial bacteria will come into the play, and they will colonize, because probiotics are an X number of cells we are going to put into the body, and that takes a while to grow. So FMT, you're actually pumping in quite a significant amount. So that is going to start colonizing it. And that brings in a balanced gut microflora restoration. The other thing is the ARGs, antibiotic resistance genes. Now, higher ARG prevalence when they are present in a human gut, when we have this transplantation, similar to the diversity of the strains, we also increase the diversity of the non-resistant genes in the patient's gut. This is how whole microbiome balance will be brought by using fecal microbiota transplantation. And these people have been highly screened and used as donors. These people who are going to donate poop every day, and those poop is collected and then processed in a very systematic manner. It may sound like poop poop, but it is actually looked at as a culture of cells, and then we go to process it and use it. And what happens in a conspecific strain competition when we introduce the susceptible strains? Now, one is we said healthy ones, right? Other ones is you are going to populate with highly sensitive strains in the FMT, and that is going to do the transplant and then increase their numbers and reduce the multiple drug resistance strains. It's such a simple, it looks like it, but the process is long, drawn, but it has been now found to be safe. So in the journey, clinical efficacy, there are enough studies have been done. They identified resistant bacteria and then administer the FMTs, then monitor, continuous monitoring of the decolonization. That is where it comes in, because observation for CRE is very important to ensure we do not introduce a resistance strain in between. And that is when we make sure the entire process completes. Premix is a trial which was done, that the name of the trial was the premix, and that showed beautiful restoration of the gut microbiota with the susceptible strains. So there are a few challenges, of course. The sample sizes are very small because you need to have X number of donors, right? How do you that requires a lot of standardization processes because it is not easy. Just like phage therapy, which cannot be just made scaled up and done. This is also such a problem. But Rebiota is doing a good job, I believe, and I hope very soon they will be much more successful and many more companies would come in similar pattern. So in conclusion, FMT is an amazing tool in addition to the small molecule and large molecule inventions that are going on, which will target and address antimicrobial resistance. Thank you very much. I hope I was in time.
Yes, thank you, Manish. An amazing talk, and yes, very much on time.
Thank you.
And we will have the discussion at the end of the session.
Sure.
I'm handing over the mic to Dr. Vasant Nagwekar, who is a senior infectious disease consultant in Mumbai and deputy managing trustee of the AMR Declaration Trust. Vasant, over to you. Let me see what Taslim Arif has joined. Manish?
Yep.
Until Taslim is joining now, you would like to add a few more points? How fast the microbiome field is developing in the field of AMR?
Yeah. I mean, I'll be glad to do that. So, one moment. Sorry?
How fast
Are there any
the
questions?
microbiome field is developing in the field of AMR control?
How fast you mean?
How fast? Yeah, the microbiome field is being utilized in the AMR control. How we
Yes,
can utilize the microbiome?
it is growing quite well in the sense to such an extent that the American Society for Microbiology has now formed what is called as ASM Health is a division, and under that they are now making encouraging people to come on board with what we say is finding to ensure that people will start using and investing money into microbiota as a tool for addressing antimicrobial resistance. Not only that, and also non-communicable diseases such as therapeutic interventions for oncology, for metabolic disorders, for IBS, etc. In all those areas, also microbiota is going to be used in the future.
Wonderful. Thank you, Manish. Dr. Vasant, Dr. Taslim Arif has joined.
Yes, so thank you, sir, and good evening, everybody. On the behalf of antimicrobial resistance, we're having a very interesting CME today on all the various aspects, beautifully crafted program by Dr. Ghafoor. And I think we'll go forward. Our next speaker is Dr. Taslim Arif Sayed, who is the CEO of CCAM Bangalore, one of the India's most influential biotech innovation hubs. CCAMP nurtures and incubates numerous deep sciences startups and also leads the innovation pillar of the National AMR Action Plan. We are honored to have Dr. Taslim Arif here, who will be speaking on innovation in biotechnology, crazy enough to work. Over to you, Dr. Taslim.
Thank you. Thank you. Good evening. And thank you so much for having me here, Dr. Kapoor and Dr. Vasant and all colleagues. I just wanted to check how much time do I have?
20 minutes.
20 minutes. So if it's okay, I'll just quickly run through a few slides to represent some of the work. Is that okay?
Yes, yes.
Okay, okay. Let me, okay. Can you see my slides?
Yes. Your slides are visible. Yes.
Okay, okay, excellent. So, no, thank you. It's wonderful to be here on this Saturday evening for this very important aspect. I think we all know the broad reason that we are here. I think it's a responsibility of key stakeholders of the community in health and beyond for us to talk about the threat of antimicrobial resistance and what we can do to minimize the damage and build new solutions to prevent further casualties for him. As indicated by Dr. Vasant, I am Taslim Arif Sayed. I work as this CCAMP as director and CEO. I hope my slides are moving. So CCAMP is an initiative supported by Department of Biotechnology, Government of India, with its mandate to enable high-end and catalyze deep science research innovations for societal impact. That's the big picture. We are an organization based out of Bangalore, where we foster deep science innovations, entrepreneurship, drive high-impact research in academia industry, and translate and commercialize academic discoveries into technology-related society. I think that is a very important aspect. As we all know, solutions are the final last mile for science impact to the society. And I think using scientific knowledge to build solutions which can address issues across healthcare, agriculture, environment is a very important aspect. So that's where my organization works. Just to give you a perspective, while we do a lot of things, one of the key aspects that we do is the enable innovative solutions. So what do we do towards it? So over last 14 to 50 years, CCAM has been supporting a lot of innovations as a mandate, a lot of research as well, but a lot of innovations, and you see a quick glimpse of it. This is a very large number and very important component because India hadn't seen such kind of trajectory in terms of building new science. So think of it. These are all new innovative biotech companies, period. Right? This is really fantastic. And this is not all. This is just a fraction of what we do. There's a fraction of number. Total is around 600 companies. While we do a lot of innovations in different domains, one of the components that we heard in today's topic has been AMR. So in 2021-22, under the guidance of the Office of Principal Scientific Advisor and the PSA himself, we set up what we called as an India AMR Innovation Hub. The idea was to converge, collaborate, and combat together against AMR. And that is the main thing where we will build innovations for, as you can see, further capabilities in stewardship, environment, human diseases, surveillance, and One Health aspects of it. So that is mainly for reasons. And what you can see on the screen is that we have been lucky enough to build a consortium at the national level, which actually brings together quite a few number of people, including global foundations, which are here, industry, academia, startups, and of course, government altogether. The Professor Ajay Kumar Sood, PSA to Government of India, is the chair of IAIH Advisory Council. As he says very clearly here, if we want to solve the huge global threat posed by AMR, India will have to play a central role at the world stage. And we are one of the first few countries globally to initiate a national action plan on AMR. And as you all know, 2 days back, we have launched NAP 2.0. Second version of AMR is out from India. So that's something we look forward to building on it. And CCAM has been clearly designated as the innovation arm of India's national action plan. But we need more cohesion, direction in our efforts. And IIH, India AMR Innovation Hub, anchored by CCAM, will play the role of a platform for convergence of mandates across the ecosystems to enable collective action. So that's the major competent and leadership message from Professor Ajay Kumar Sood to build this IIH together. And of course, when these are some of the members who are there, as I mentioned, idea is to bring all of them together and see if we can solve address the India's prioritized AMR problems altogether. So vision is to have a build targeted innovative solutions for India's top 10 AMR challenges across human health, agri-animal health, and environment health. That is across One Health for India and beyond during the period of NAP 2.0. So NAP is launched now. So we are looking at the period of 2026 to 2030 to build those solutions. Of course, the domains remain prevention, screening, diagnostic, therapeutics, antibiotics, surveillance across human, agri-animal health. And idea is that IIH will here fund those solutions, handhold those innovations, and help with deployment and implementation of this innovation. So it's almost prioritization, funding, handholding, and deployment. So it's all the entire cycle through our partnerships with different partners who are at the IIH level and beyond, of course. And we have international partnerships who are there, who have been supporting us through various things. CARB-X, which is the world's largest funding aspect for the AMR. We have GAMRIF from UK as our collaborator. We have collaboration in Denmark from ICARS, collaboration with SwissNext. Netherlands, we have collaboration. And of course, SBI Foundation is supporting us for this. And of course, we have been CARB-X, as some of you would know, CCAMP is only one of the 4 global accelerators supported by CARB-X, the other 3 being either in the North America or the Western Europe. So we are very proud to be in this senior league of global accelerators for AMR. And that also gives us a significant connect for us to take some of our innovations forward. The 3 innovations which are supported by CARB-X in, if I'm not wrong, Asia, but for sure in India, all of them are from CCAM. So that 2 are in therapeutics, 1 is in diagnostics, that is Bugwork, Gangajan, and Module. At the same time, of course, we are supported by SBI Foundation, where we are supporting directed solutions for neonatal sepsis, UTIs, in One Health, bovine mastitis, aquaculture, poultry, and of course, at the late stage of it, and a lot of work has gone in and significant kind of good work has come out of this. We also have collaboration with GAMRIF, where we are supporting AMR in environment. So the CARB-X is for human, SBI Foundation is for One Health, and GAMRIF collaboration, where total around nearly, if I'm not wrong, 60 crores we are putting in for this program altogether over 3 years that we have started now. We are hoping to do work in terms of addressing the issue of AMR in environment, and I will be happy to talk about some of the work that is there. And of course, ICARS, we have just began in One Health aspect as well. So there are 4 different programs, friends. I just want to say the entire aspect of IIH is to enable innovation. So we have 4 programs, separate major programs going on together, possibly. These are nearly, I'm trying to believe that this is almost 80 odd crores we are putting in already, and we are hoping to add more to see what we can do in AMR innovations. This is just starting, so we are hoping to add more numbers to that across these 4 programs, and we are hoping to build more programs. Just to give you a quick perspective of kind of innovations which are doing, this is in therapeutics, Bugworks, most of you have heard about it, but the world needs new antibiotics. I think we all know it, and I don't need to underscore that at all. I think it's very much And Bugworks out of Bangalore at CCAM is in the world the most advanced asset for broad spectrum antibiotic. Phenomenal work. They are in the phase 1 trial in Australia, and we hope that they're able to then address the discovery void, which is there for almost 35 to 40 years in the space of broad spectrum antibiotics. So we are very, very excited this work coming out of India. At the same time, so this is some of the component that Kangijan is working on, which is phage derived peptide. Another approach in therapeutics. Again, they are heading towards the clinical trials. In medical devices, we have Koyo Labs in Excel, which has US FDA approved device for prevention of ventilator-associated pneumonia. We all know the issue of VAP at in ICUs when patients are ventilated, and unfortunately, many of them acquire pneumonia, which can be fatal. This is a very smart medical device that prevents secretion of oral saliva, oral secretions. It prevents it to go to your lungs and prevents the occurrence by almost 90 to 95% of occurrence of VAP. This is really phenomenal work that Koyo Labs and Inexcel has done. This is already in market, USA FDA approved in many countries now there. We have Module, which is trying to build ultra-rapid detection of bacteria in UTI and also doing the resistant profile. Really exciting work. They have validated the clinical samples, and they are now launching it soon enough to take it forward. We have come. This is now prevention. So if you can see, it's a very interesting profile in human health itself. Biomonitor just got 3 days back, they got US FDA 510K approval for their work to prevent spread of drug-resistant infection in healthcare environments, including ICU, which are multi-bedded ICU. How can they make the entire air health very sterile in those settings with more than 99.999 efficiency? And they have now data from hospitals on reduction in occurrence of infections when you actually deploy this innovations or solution in hospitals. So this is really important in terms of prevention of infections and resistant infections broadly. At the same time, we have companies like Haystack, which is doing fantastic molecular genomics work to test the neonatal sepsis. Again, they are doing very good work for gene sequencing based, already using Oxford Nanopore technology. Achira Labs, which is doing microfluidics patterns for UTI detection and antibiotic susceptibility. We have work for a simple disposable paper article platform for childhood diarrhea, differentiation of childhood diarrhea. Again, very good work. We have ASIA Healthcare for using local antibiotic rather than systemic antibiotic to prevent the broad exposure of AMR upon root canal treatment or many others. We have CRISPR bits, which is doing fantastic work for diagnostics, including the point-of-care test for 5-carbapenem resistant genes, APC, NDM, IMP, VIM, and OXA48. Really fantastic. It will be almost an LFA, like a pregnancy kit, which could be the readout at the end. So this is really very good work. This is happening for diagnostics, and you move quickly to animal health and mastitis, which causes a lot of use of prophylactic patterns of usage, as well as heavy dosage of antibiotics without knowing what it is. This allows a phytotherapy for mastitis, which is already at the clinical stage for bovine. Similarly, another thing for mastitis is for prevention, where you can actually do a simple TID solution post milking to actually ensure that there is no further infection patterns that emerge. Because after milking, the skin is kind of affected and ruptured, and that causes a lot of infection as well. So post milking, when the skin is hurt, how can you actually provide the solutions to prevent infection at that time? Again, mastitis, we have several, because there's a huge issue. We have simple quad test, which is almost like a small cup where you can actually put milk and figure out the subclinical mastitis detection. Again, this is in market. These are not promises, friends. These are things in market already used by thousands of farmers, animal farmers, and allowing them not to necessarily use prophylactic use of antibiotics in animals. Again, many other things, which is, in aquaculture, how do you ensure that you provide fish meal which has inherent indigenous antimicrobial peptides, so that the aqua farmers, when they're doing shrimp, they don't have to do prophylactic or strong use of antibiotics, because if fish meal itself, which is coming from the insect culture, has the antimicrobial properties, it can prevent infections and hence prevent use of antibiotics, which is very heavy, which is causing a huge damage to aqua farmers because of the residues that stay back. Some of the other companies which we have supported under the environment are like FNDR, which is treating hospital waste and taking away the antibiotics, antibiotic residues, and other bacteria and resistant bacteria. Very interesting work they are doing. Biomonetize developing air microbial index for the first time. Again, very good work, and many others which are happening. In the interest of time, I will start slowing it. This is the work that they are doing. FNDR, this is the air microbial index. Some of the workflow that is happening in detection at the point of care. I'll skip a few in the interest of time, but a lot of this innovation, these are all individual innovations, friends, that is happening. And you can imagine a lot of work that is happening to prevent damage of AMR in human health, animal health, agricultural health, and environment as such. Just to close on this, so outcomes is that over the 5 years, we want to build at least 10 to 15 innovative solutions addressing top 10 AMR problems in human health, animal health, environment. And you can see we are already in a good stage. We are already building some of the fascinating work, and with collaborations with friends like you and others, we can actually make it really solid. I'll close by saying it that we are building a path. I think India is at a very important stage, as we all know, as you saw the quote from Professor Sood. India has the capability to build indigenous solutions, which are important for us, but these solutions have capability to address issues of the rest of the world, including Asia and Africa. So great to have you, join this thing. Thank you for the opportunity and looking forward to interactions. Thank you so much.
Thank you so much, Dr. Taslim Arif. I think there's a lot of encouragement from your side, your team. And I think we, in antimicrobial resistance, we were thinking that we are lost. But I think listening to you, there's so many innovations, not only in humans, but animals and even agricultural fields. I think it's very, encouraging. So many things, not only in diagnostics, but even I'm seeing that so many things coming up into therapeutics. Of course, diagnostics is the beginning towards reduction of antimicrobial resistance. It is diagnostic, therapeutics, and newer drugs, and especially alternatives to drugs like patch therapy and all that. I think you're all doing a fantastic job, and I think we really look forward to see all these innovations coming up in next 3 to 5 years. I think this will help and lead India to reduce what the world is talking about. India is having a lot of AMR, like the Lancet paper which recently came. I think will definitely all your ideas, solutions, innovations will help India to progress further. Thank you so much, Taslim Ali, for the wonderful talk, and we'll have questions in the end. Thank you so much. So
Thank
our
you.
next speaker, thank you. Our next speaker is Dr. Amani. Yeah, I think Dr. Amani.
Yes, Dr. Amani is joined. Yes, Dr. Amani is
Dr.
joined.
Amani Al-Nimir, she's going to speak about flipping the script. What can be the AMR game changers? And she's speaking to us from Saudi Arabia. So we welcome Dr. Amani, microbiologist and infection control consultant from Saudi Arabia, well known for her leadership in innovation, entrepreneurship, and her strong engagement in the antimicrobial resistant education. And thank you for joining us, Dr. Amani, and we look forward to hear from you. Over to you, Doctor.
Yeah. Good evening, afternoon, and morning, everyone. Can you see the slides?
We can see your slides.
All right. Okay. Yeah. So it's a pleasure to be here. Thanks to Dr. Ablafour and his team for the invitation. And I mean, the great thing about being here today is to talk about really bold and crazy ideas. And if we don't think crazy, as just Dr. Ablafour started by saying that the antimicrobial resistance evolution will be, like, I mean, ahead of us. So what I'm going to talk today is about, like, I mean, how we're going to flip the script in relation to the prevention at its very roots. And here I'm talking about vaccination. So vaccination against AMR, and I'll be presenting two different approaches about the vaccination in relation to the antimicrobial resistance. So, I mean, all what I'm going to present is based on the traditional vaccines that we know that they help with antimicrobial resistance, because we know they lower infection, then less exposure to antibiotics, less selective pressure, and then you have less resistance amplification. And the data from vaccinations, for example, like pneumococcal vaccine, Haemophilus influenza B, and others have shown that more than 100,000 deaths are prevented annually, according to the WHO data. And not only this, that would reduce also the selection for antimicrobial resistance in our microbiome. So if we want to build on that concept, then we think about AMR vaccines. And then there are two ways to think about AMR vaccines. We can think either of one very bold idea, that is to tackle the resistance determinant itself, for example, a plasmid in gram-negative, but that's still, based on the current science, far away from implementation. That's a very bold idea. It's open for research, but as I said, it's far from implementation. But what I want to focus on is how to train our immune system to recognize and destroy multi-drug resistant organisms through vaccination. And I'll be using ESBL as an example here in this illustration. So I'm not focusing on the AMR itself, but again, as I said, on the bacteria that harbors the AMR. So just a quick refresher. So plasmid is that self-replicating DNA material in the bacterium, and it can be present in any many organisms, and most of the gram-negative resistance determinants that we are suffering from today are carried on plasmids, ESBLs, CREs like NDM, KPC, and others, as well as MCR14 resistance. The theoretical model for developing vaccine for the plasmid itself will follow just like any other vaccine model. So you will develop a design for the vaccine, pick up a target, which could be either a conjugation, for example, a protein target, or other bio-domain target from the plasmid work. And then you will be studying it in the different phases of vaccine development. And as I said, this is a model that is still described as a potential, let's say, direction for tackling AMR, but there isn't much progress that is happening in that particular area. But one can, especially now we have a lot of genomic data, AI tools are available for molecular docking, and then you can develop your design of the target, and then the testing can happen. However, in my opinion, this is not a good, let's say, target, because if we are talking about a plasmid or resistance determinants, then you are talking about something that is really not accessible to the immune system. It's deep within the bacterium. It's highly variable, not conserved. So even if we manage to develop a vaccine, let's say for one of those plasmid, then definitely the escape phenomena will be encountered very soon. So the second model that I want to focus on is one where we tackle the bacteria with characteristic AMR determinants. In other words, we are targeting that plasmid containing pathogen, and we are developing the vaccine against it. And if we talk the ESBL as an example, so the first thing we want to to highlight the enzyme itself, or the ESBL beta-lactamase itself, is not a good target for vaccine. It's highly variable, it's continuously changing, and it's different from geographical to another geographical area. And again, it's not accessible for the immune system. So it won't be a good target to develop vaccine for it. However, if we look for other proteins that are accessible on the bacterial surfaces, and they have been well associated in research with the plasmids carrying AMR resistance determinants in gram-negative, and in particular, I will focus on siderophores receptors, for example, the iron, So in ESBL in particular, there's a good association between those receptors and the plasmid carrying resistance. And because these receptors are accessible, are on the exterior surface of bacteria, so theoretically speaking, they can work as a good target for vaccination. And the good thing about them is that they are conserved across ESBL strains, at least in one specific geographical area or one ESBL genotype. So it's just we'll follow the normal step-by-step concept for developing a vaccine, where you pick up a target and then you start by the cloning and the mass production and then testing it, starting like, in vitro, ex vivo, in vivo animal model, and finally, if it works, in a human. So you'll be working to test efficacy as well as safety. And if we can test that concept and if we can optimize it and deploy it, then that can be a One Health solution, because we are not only preventing infection by MDRO in human, but also we are reducing the reservoir, the environmental reservoir, as well as the animal MDRO burden. So it's a very bold idea. It's in the early area of research, but I wanted to share it because I felt like with the AMR grants that are coming, looking for innovation and all of these bold ideas, these type of work need multinational collaboration, and they need multiple brains to sit together and experts from different background to prepare a good proposal for such work. So of course there are challenges are ahead for this work, but challenges are never barriers for innovation. So although we said, for example, the CD4 receptors are conserved more or less, but again, we will deal with some diversity, especially when we are talking about various geographical areas and different populations, and the escape phenomena. And you want always to maintain the microbiome balance and avoid the dysbiosis, and obviously all the regulatory constraints and difficulties. However, I still believe that building, let's say, the first vaccine that can prove at least ex vivo, not necessarily even in vivo, that it will work, it can be the nucleus for an ecosystem vaccination at scale. It can build a network for resistance containment in the future. And on the long run, it will complement antimicrobial stewardship and new drugs in the pipeline. And obviously, it will be a new class of AMR vaccines. So I just listed here some references that keep in line with this direction of thinking for vaccination. And just thank you for your attention. And I would like to get this opportunity to invite you all to join me on my podcast, the Medics Podcast, in particular, because I'm going to host an episode in January with an expert in plasmid vaccines, who is Professor Margaret Liu from University of California, who has good experience with building plasmid vaccines. And we hope that out of this episode, we can come up with some collaboration. So thank you, and happy to take any of the questions.
Thank you so much, Doctor. And I think this is again a new innovation. I think I had not heard about vaccinations against antimicrobial resistance. And I think with this blocking the cidofovir, and I think this is something new to learn for us. And we'll have questions for you at the end of the session. Be staying with us. Sir, Gafur sir, do you have anything to say? I think vaccination against AMR.
Yes, it's an amazing concept. Some work has been going on, but listen to Dr. Amani, including the plasmid vaccines. It will be amazing to have these vaccines against drug-resistant bacteria. And as Amani has mentioned, may not be really the preventative or probably partially therapeutic. Where patient is having recurrent CR Klebsiella UTI, it will be amazing to have a vaccine. We can prevent recurrent infections. It's an amazing concept.
Right. Thank you so much, Doctor. After this, we have Dr. Arvind Regukumar, who is a good friend of mine, and he's going to speak about the upside down approach to stewardship. And he's heading the infectious disease department at Government Medical College, Thiruvananthapuram, and the lead of the Kerala State Antimicrobial Resistant Action Plan, the state in India which is leading against antimicrobial resistance. Thanks, Arvind, to take that lead for government. of Kerala, and is the first and most successful state-level AMR programs in India. Welcome, Arvind, and we look forward to listen from you.
Thank you, sir, for the kind words. At the outset, I would like to thank AMR Declaration Trust, APUA, especially Gafu sir and Vasan sir for this opportunity. So, are the slides visible, sir?
Yes.
Okay. So, after listening to three brilliant talks prior to this session, I feel like a rank outsider because I'm not going to talk about any path-breaking stuff or research or something like that. Instead, I would like to take you through the approach that our state adopted in the implementation of its state action plan, that is Kerala Anti-Micro-Resistance Strategic Action Plan, which was the first state action plan to be released in India way back in 2018. Now we are into the 7th year of our action plan. So why did I title it as upside down approach to stewardship? The basic reason is that the rate of AMR varies across the different geographies. So we know that Southeast Asia is actually the hub of AMR, as well as the maximum gamut of AMR related deaths and all. And the AMR rates differs between the developed nations as well as the developing countries, so much so that it has been titled as a socio-economical problem. And anywhere in the world, like during the UN General Assembly meeting on AMR as well, it was one thing which was raised is that the lack of political commitment. So how can we get, the politicians also totally involved in the AMR strategy? Something I think that I will take you through the next 10 minutes or so. So basically, these are the 6 strategic priorities in the Global Action Plan, National Action Plan, as well as the State Action Plan. So the first, the 4 of which are core academic priorities. There is awareness and understanding, knowledge evidence, IPC, antimicrobial stewardship. 50 is with regard to research, and 60 is obviously collaborations. So when we talk about antimicrobial stewardship or the research on that, we always think that this has to be led by a clinical microbiologist or an ID person or a clinical pharmacologist, etc. But if you're looking at the success stories from India, so COVID, how we managed COVID can be termed as a public health success story because of the all of government approach. So all the arms of the government was actively involved in the treatment of COVID, as well as how the vaccination got rolled over, etc. But when you are looking at AMR, is government working like that? We know that we have in One Health the concept, and all AMR is a One Health issue. But are we adopting the same? All of the government approach in AMR, I am a bit skeptical at many regions. Okay, so when we go through the data from Kerala, so 1 month back, 4th antibiogram. So that is every year we actually publish the state antibiogram. So this represents the data from 55,000 isolates. You can see that there is a slight decline in this is the carbapenem resistance rates. Acinetobacter from it is around 45% resistance, E. coli 6%, Klebsiella 26%, Pseudomonas 17%. This is actually data from 2018 onwards, and probably I don't think that we should be paying attention to data in 2018 and 19 because at that time we did not have that much of labs in our network. So you can see that there is a steady decline in the carbapenem resistance rates in comparison to the data from the ICMR network as well as the national network. You can see that the carbapenem resistance is maybe around, say, 30 to 40% lesser in the priority pathogens in Kerala. And the most interesting aspect is, we have our district antibiograms, that which reflects the resistance from our the Taluk hospital as well as the secondary tier hospitals. So this is a district antibiogram from Ernakulam district, which was released last year. So you can see that this is the SPOCS means the data which flows to our network from the secondary care levels. The carbapenem resistance in the secondary care level is actually negligible when compared with the state data, which reflects the resistance in the tertiary care level. In the secondary level as well as the primary level, we don't have any carbapenem resistance. So here actually gave us an idea, like when we are talking about this tertiary care centers, and we are actually talking about antimicrobial stewardship, we have 3 pillars, right? Diagnostic stewardship, IPC, and antimicrobial stewardship, and we are working in the ICUs where the carbapenem resistance is pretty high. There I will put the priority more as IPC, diagnostic stewardship, and antimicrobial stewardship, because carbapenem resistance is there, then basically it is about using the rest of the drugs in a appropriate fashion. So in a tertiary care center, the priority definitely will be on preventing an infection from occurring. But whereas when you are looking at the secondary as well as the primary level, so there a huge opportunity is there for antimicrobial stewardship. So in our action plan, instead of starting all these stewardship activities, definitely the stewardship activities will happen to an extent in the tertiary care center because of the access to clinical microbiology, ID or trained medicine people, etc. But in the secondary and in the primary care level, no one talks about stewardship because there we don't have any trained microbiologist or the ID person or people who are really trained in stewardship and all. But we decided actually to focus first on the primary and the secondary care level. We realized that the real opportunity lies there. Because, if you're looking at the stratified antibiograms, there, as of now, we don't have that much of resistance. So if you can target the stewardship activities at the primary and secondary level, then probably you can bring down the community, the resistance to the priority pathogen sort of thing. For that, we actually wanted everyone in the Kerala to be aware of the basic concepts of antimicrobial resistance. For that, we started this innovative campaign way back in 2019, that is Antibiotic Literate Kerala campaign, with 4 objectives. So all these objectives were aimed at educating everyone in Kerala about the right use of antibiotics. That is educating the farmers, the civil society, the students, the pharmacists, doctors, all healthcare workers about the right way in which this antibiotic should be used. And the campaign is supposed to end by the end of this December. Okay. So how will we take this the message to the root level? So someone sitting in a tertiary care center may not be able to convey this message to the farmers and all those things, or the public and all. For that, actually, we came out with this district AMR committees. So in our state, we have district AMR committees which are established 4 years back. We have 14 districts. Then we established the 14 block level AMR committees. We have around 190 health blocks, so 190 block level AMR committees. And we have this different SOPs for how this district AMR committee should function, block level AMR committees should function. And based on this, through the block level AMR committees, we will engage with the panchayat, that is our ground level local self-government bodies, and through the panchayat will educate the community at large. So this was the concept. For this, we came out with the concept about antibiotic smart hospital initiative, which is actually a very early model of participatory antimicrobial stewardship. You all know that in around 2 years back, that is in October 2023, WHO came out with this concept about participatory antimicrobial stewardship. That is, we need to go beyond the hospitals and we need to engage with the community, and they should be taught about the right IPC and all those stuff, how to use antibiotics, especially in India where this OTC sale of antibiotics is so rampant, etc. So participatory stewardship forms the cornerstone of this antibiotic literate state concept, etc. And this the stewardship part was written onto our action plan way back in 2019 itself. These are the 4 pillars of this antibiotic smart hospital initiatives: prevention of infection, access to essential health services, timely accurate diagnosis, appropriate quality assured treatment, and all. Basically, it's a quality initiative. So basically, when we talk about AMR, in many settings, it seems like an abstract thing. So the most important thing as far as any action plan is concerned is that we need to have a monitoring and evaluation framework, like what we have in HIV or what we have in TB. For AMR also, we need to have a very robust monitoring and evaluation framework. That is what we actually did. And we established this criteria to declare hospitals as antibiotic smart at the primary care level, secondary as well as tertiary care level. At the primary care level, so they satisfy 10 criteria, we'll declare them as antibiotic smart. 2 criteria are pretty difficult. One is that WHO now says that by 2030, 70% of antibiotic use globally in a country, in a state, or from a hospital should be from the access category. So if we want to achieve that target, so 70% of antibiotics to be from the access category, that means that in the OPD, more than 95% in primary care, or 90% in secondary care, or 85% in tertiary care should be from the access category of antibiotic. The second difficult target to achieve is actually that the IPC aspect is taken care of by looking at whether the hospital is having some sort of a certification. Could be an NQA certification, Kaigal, Venebeja, sort of things like that. So once we actually declared a couple of hospitals as antibiotic smart in the primary level, then we came out with this criteria for secondary and tertiary care level. And there the criteria are a bit more stringent, like we look at the surgical safety checklist, prospective audit and feedback, low-hanging fruit model of stewardship, the high surveillance, and also the environmental dimensions like whether your STPs and ETPs are as per the guidelines from Pollution Control Board. So basic idea is that by establishing these antibiotic smart hospitals at the panchayat level, that is at the primary care level, you are actually engaging everyone in the panchayat to be part of all these stewardship activities. So you are making a society aware of at least what AMR is and what they are not supposed to do. Like you should not be taking drugs without the prescription of a doctor and all. So this sort of, over the last 3 years during this awareness week and all, we have been conducting activities across the state in all the health blocks, in all the panchayats, etc. It's like a mass movement or an all-of-government approach. The politicians at the ground level are totally involved with these activities. So that, a couple of years back, around 2 years back, January 1st, 2024, when the state came out with this Operation AMR, that is, the total ban of OTC sale of antibiotics in the state without prescription. Okay, then there was no opposition to that. No, the media did not come out against that, and so everyone accepted that because because of the prior work done by creating awareness among the public. Suppose someone is able to get an antibiotic without prescription from a pharmacy, they can use this toll-free number and register a complaint with the drugs controller. So then an action will be taken against that offender, like that. So these are the criteria to be met in the tertiary care centers. And following this AMR program, so you would have read in newspapers that there is a decrease in the sale of antibiotics by around 25%. That is, it's around 1,000 crore per year. So that has happened just by a regulation. Now the next aspect is that making a hospital the primary or a secondary care level as antibiotic smart is just the beginning. The next step is that we all know it's a One Health sort of issue, and then they are supposed to do this AMR hotspot mapping. So this is done by a family health center in Tagari in Alapi district in Kerala. So all the FHCs are supposed to do like that. They will map the AMR hotspots in their panchayat. So the AMR hotspots in this panchayat are, one is that there is a fish farm, the hospitals are there, then poultry farms are there, then the pharmacies are there, other hospitals are there, doctors are there, the schools are there. Schools means you need to educate the children, etc. So they will come out with micro action plan along with the animal husbandry department, etc., pertaining to this panchayat, so that they will go for targeted IEC sessions in all these AMR hotspots, and that is how we plan to make this panchayats, etc., as antibiotic literate. And a criteria has been we have come out with a criteria for the same. And then this was something innovative, which was, a pharmacist gave us this idea that is antibiotic should be dispensed in blue cover. So why in a blue cover is that we know that the blue is the color of, so go blue for AMR. That's the WHO campaign during World Antimicrobial Awareness Week every year. So usually all the drugs from the hospitals are dispensed in white covers. So, and we give advice like this antibiotic should be used like that, this like that. But unfortunately, in the same cover, you are having this, the drugs for the paracetamol, then cetirizine, this anti-diabetic drugs, hypertensives, etc. And the poor patients, illiterate patients may not be knowing what an antibiotic is. So we implemented in the state that antibiotics should be given only in a blue cover, and the blue cover will be having IEC messages. So these IEC messages will be like, so how there are 4 messages on the right use of antibiotics. So just by changing the color of this, the cover and giving antibiotic in a separate cover, we were able to convey the message that antibiotics are very precious and should be handled with care. And then the next aspect is addressing the environmental dimension of AMR. So what will you do with an expired antibiotic or antibiotic which you are not going to consume in future? Usually it used to be disposed in a landfill or water body. So as part of this PROUD program, so PROUD is a program for reuptake of unused antibiotics. So we can keep these bins in the nearby the government hospital, so that the people in that panchayat are educated to give this date expired antibiotic back to the hospital or the pharmacy. So this is also one this criteria to be satisfied to be declared as an antibiotic smart hospital at the primary care. That's keeping this box as part of the PROUD program. This is an example of participatory stewardship in action. So you can see that a nurse from a primary care center is going to the household and educating the public about the IPC practices, as well as creating awareness about the right use of antibiotic drugs. That is, participatory stewardship and making a Kerala antibiotic literate campaign is going on. So the tagline for the campaign is My Kerala, Antibiotic Literate Kerala. So, and then we actually divide the hospitals in the state as per the criteria achieved. So if you are achieving 15 criteria, so you'll be having a light blue color, then you will be termed as an antimicrobial stewardship compliant hospital. If if the color is in the dark blue, it will be an antibiotic smart hospital. So we'll know where these hospitals stand with regard to the criteria achieved at the primary care level and secondary care level. Even if if the color is the pink to begin with, it does not matter, because then you need to have a micro action plan so that you can move up the ladder in the next 6 months or so. And then this year, the good thing which has happened is that usually NHM gives awards, the Kayakalp awards for IPC and all those things. This year, the antibiotic smart hospitals were also given awards. So you can see that the entire hospital, all the staff coming together to receive the award. So sort of an ownership is shown by the panchayat. The panchayat members are coming to receive the award. So if the panchayat members are coming, means that the local politicians are also involved in AMR control activities. So that is the biggest thing that we can do, getting everyone in the state engaged in AMR control activities. And this is about the process metrics. Since we have stipulated that 95% of antibiotics in a primary care hospital should be from the access category, and this auditing is done every 3 months, and we have an idea about the use that is happening in the different hospitals in the. This is an example from Ernakulam district. And just to remind the doctor that they should be using more of access. You can see that in front of the doctor there is a calendar. There's a green thing is the access drugs, yellow is the wash drugs, and the red is the reserve category of drugs. So what impact that all these activities has done to antibiotic prescription pattern in Kerala over the last 7 years? So the use pattern is actually tracked by World Bank through this Pharma Rack, which sort of gives an idea about what is happening in the private sector as well as in the government sector through the secondary distributor level data. So you can see that from 2018 to 2023, there has been a 13.3% decline in the sale of antibiotics in the state. Mind you, this is actually prior to the regulation, that is AMR program. So prior to that itself, there has been a 13.3% decline in the sale of antibiotics in the state. The access usage went up by 1.8%. The watch antibiotics prescription showed a decline by around 24% in the same period. And WHO says that the access usage should be to the tune of 70%. You can see the state average is around 58% now. So this sort of tracking is happening in the state. So what are the conclusions? These conclusions actually were made by a PHC which has implemented this antibiotic smart hospital concept. So their conclusion is that a smart PHC is not just a place to treat illness, but a hub for preventing antimicrobial resistance. By adopting stewardship at the ground level, a PHC can safeguard antibiotics for future generations, reduce treatment failures, ensure availability, accessibility, accountability in antibiotic use, empower health staffs, community, and other departments, private hospitals, etc. So basically, all these components are very, very important in going forward, so that the prediction for 2050 can be averted. IPC, antimicrobial stewardship, research, diagnosis stewardship, vaccination, establishing a bacteriophage library in the state. That aspect is also being taken care of by Institute of Advanced Virology in the state. So all these things can be taken forward in a seamless fashion, provided we have enough support from the politicians as well as the bureaucrats. So Kerala actually, the biggest thing has been that since we made it a all of government approach towards dealing with this problem of antimicrobial resistance. So we get support from all the policy makers, and that helps us a lot in implementing all these research activities and ground level implementation. So thank you very much. So this is actually the tagline for our campaign to make antibiotic literate by January 2026. That is My Kerala, Antibiotic Literate Kerala. And this capsule is our mascot. So thank you very much for the opportunity.
Thank you, Arvind, for that wonderful talk. And I think Kerala, I think other states, Kerala has done a wonderful job in antimicrobial resistance. And I think it is other states in India to follow Kerala, the same model, I think the way to reduce the antimicrobial resistance. And I think everybody talks about antimicrobial resistance and antimicrobial stewardship in the tertiary care centers. But I think Kerala is the state where, Arvind, you all started from primary and secondary, which is the most important part. Why are we talking about tertiary? There already exists AMS and AMR in tertiary care centers. But what we require is at the base level, primary level and secondary level. I think that was the way forward. And you already clearly showed that there was a significant reduction over a period of 2-3 years by prescriptions and even the antibiotic usage by almost 24 to 25%. That's a significant reduction in AMR, and I think I don't know why, but all the states in India, I urge the governments of each of the states to follow this Kerala model. At least stop the over prescriptions at the chemist level. That has to stop at each and every state. I think that is a way forward for everybody. Thank you, Arvind, for opening our eyes, and for everybody in India. I think our next speaker is Dr. Vasanti Ramachandran. She is a senior scientist at Bugworks Research, an Indian company developing globally recognized antibiotic molecules. Bugwork represents India on the world stage, and we are proud of their pioneering work. Welcome, Dr. Vasanti. And Dr. Vasanti Ramachandran is going to speak about killing two birds with one stone, Mission Impossible. And I'm eagerly waiting to listen to Dr. Vasanti. Over to you, Dr.
Is my slide visible?
No, 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 no. You
Not
have to
yet.
share your slide. There's a green arrow there. You'll have to click on that. At the base of the computer. There's an arrow. Yes. Correct.
Is it visible now?
It's visible. Just make it full screen.
Yes. Okay.
Thank you so much.
Thank you. Thank you so much for the kind introduction. Good evening and good morning to everyone. At the outset, I would like to thank the organizers for giving us this opportunity to present this work. My title definitely looks very interesting, and what it is all about, we will see it through the presentation. We at Bugworks actually are focusing towards two key areas. One is antimicrobial resistance, and the other is combating hard to treat cancer. Our vision is to become one of the most impactful therapeutics company globally. Also, in terms of our purpose, we would like to improve and extend patient lives using breakthrough technologies, novel access models, and affordable medicines. As Dr. Taslim had earlier mentioned, we have been very fortunate and have been benefiting quite a bit in terms of enjoying the fantastic ecosystem that CCAMP has offered us throughout this research that we are doing at Bugworks. Looking at what happens in a research dealing with antimicrobial resistance, it is multidimensional. In order to find solutions to combat antimicrobial resistance, it is very important that we look into multiple aspects, the prime one being looking at novel targets. It's very important that the target is novel, because otherwise we would face cross resistance, and it would be of absolutely no utility if you look at targets which have already been exploited before and against which resistance is already prevailing. It's very important to have a robust discovery platform, because that gives us the confidence of the discovery of the molecule that you are looking at. It's also important to keep up with the technology, and also very important to have a multidisciplinary team, because that helps us to progress the molecule in multiple various dimensions. That is very important for the discovery and development of a novel antibacterial. As earlier speakers had mentioned, it is also important to have collaborations and partnerships, because it is very difficult to progress a molecule on your own. So collaboration and partnerships bring in other expertise and disciplines which will help you to take your molecule forward. It's also important to have a differentiation in your molecule, because that is what would give you the market value, and also in terms of a life for the molecule, in terms of longevity, and it would bring in the necessary differentiation from the ones which are already existing in the market. And for a small biotech like us, it's very important that we have to be highly efficient because time is money. So, looking at what goes behind the discovery of an antimicrobial, there are a number of mechanisms through which a bacteria actually develops resistance. Some of it include modification of the target or degradation of the target. And lastly, but not the least, it's the efflux phenomenon. Most gram-negative bacteria have this efflux pumps built into them, which helps them to pump out the antibiotics from the cell, leaving behind very little bit of antibiotic within the cell, and that helps the bacteria to resist the antibiotic and survive without any problem. So it is very important that you dial in the while you dial in the property to inhibit the target of interest, it's very important to dial out the efflux property. And for this case, we actually built the elute platform with the help of which we were able to retain the molecule within the cell at the same time allowing the inhibitor to bind the target of interest. So what we observed is in the case of multi-drug resistance, there are efflux pumps which belong to the R&D family made up of ACRB, ACRA and TOL-C, which are the ones which help to transfer the molecule from the cytoplasm to the periplasm and finally through the channel into the outside of the cell. Now, what we found is in the case of the ACRB, there is an aromatic cage made out of multiple phenylalanines and tyrosine, which actually binds the molecule, be it an substrate or an inhibitor, because of which this recognition then helps them to pump it out of the system. So irrespective of this, in case your molecule binds to the target, it is of little use because in spite of binding to the target, the molecule is pumped out of the bacterial cell. And as you can see in the bottom of the slide, the wild type MICs is 0.25 microgram per ml, whereas in a pump knockout, it is 8 nanogram per ml, clearly indicating that in spite of binding to the target of interest, the molecule is thrown out, and that is why you see a 30-fold difference between the wild type and the pump mutant. In order to uh, combat this mechanism, it was very important to ensure that your molecule is not recognized by ACRB. And this is what we called as a stealth strategy, because of which the molecule, even though gets into the cell, is not recognized by the efflux system and it is able to bind to the target of interest. And in doing so, if you can see the MIC is given below, the difference between the wild type and the pump mutant is only of the order of 4 times, clearly indicating that your molecule is able to bind to the target of interest, but it is not being recognized by your efflux pump. Clearly, it is avoiding the pump recognition. So when we went on to screen a library, we were able to identify one of the molecules, and this molecule resembles the novel bacterial topoisomerase inhibitor structure, and clearly showing that it has a dual target, that is gyrase and topoisomerase. And as you can see in this slide, the inhibition that you can see between the gyrase and the topoisomerase is of the order of 4 to 5 nanomolar up to 9.6 nanomolar. As against ciprofloxacin, which is also known to share the same target but does not have so potent an IC50 as the 977, which is our lead molecule. In this case, it is of the order of 250 nanomolar and 11 micromolar, clearly showing a difference between the two molecules. When the mechanism was looked at, again it showed the 977, which is the gyrox molecule, actually stabilizes single strand breaks leading to the inhibition of the bacterial cell, and there is accumulation of nicked DNA as against ciprofloxacin, which stabilizes double strand breaks and leads to inhibition of the bacterial cell. So clearly what we have brought into this molecule is not only in terms of isopotent nanomolar IC50s, but also in terms of differences in the mechanism of action, clearly indicating that though they both inhibit the same target, but there are differences. We had also found out through structural studies that the interaction residues between the two molecules are also different and they are spaced apart. Because of the lack of efflux activity, as well as having this potent inhibition on these two essential enzymes, which are gyrase and topoisomerase, we were able to get very potent MIC90s against many gram-negative bacteria, be it aerobic or anaerobic, as well as gram-positive pathogens. And as you can see, most of the MICs are sub-microgram per ml, only up to only in terms of Klebsiella going up to 2 microgram per ml. This molecule is also effective against biothreat pathogens and anaerobes. When you look at the MIC pattern between the sequential isolates and the resistant isolates, the pattern looked very similar, clearly indicating that the presence of any pre-existing resistance did not have an impact on the MIC, and the MIC90s were clearly potent and similar, be it sequential isolates or resistant isolates. We were able to screen almost 10,000 isolates, which we were able to access through different agencies as listed here, clearly indicating that we have a broad spectrum antibacterial with potent activity specifically against the gram negatives, which are very difficult to achieve. And as I had mentioned earlier, the mode of action of the 977, which is our lead molecule against ciprofloxacin, since the mechanism was different, we were also able to demonstrate the lack of cross resistance, clearly showing in this picture against Acinetobacter, Pseudomonas, Klebsiella, and E. coli. The 977 MICs were very potent, even though each of these isolates were resistant to fluoroquinolones. Thus, there was a clear indication of no cross resistance between fluoroquinolone and 977. So while we were optimizing the molecule in terms of its antibacterial activity as well as the target enzyme activity, it's also very important as you progress the molecule to look at other characteristic of your molecule, specifically in terms of secondary targets, which could lead to some toxicity. Now, it has been known in the field of novel bacterial topoisomerase inhibitors that they turn out to have hERG inhibition, due to which there are implications of cardiotoxicity. So as we progressed this molecule trying to improve on the bactericidal properties, we also wanted to see whether we will be able to get rid of the hERG liability. And this is what is clearly obvious in this case, that as soon as we were able to get the lead molecule, the hERG liability also drastically reduced while retaining the dual target and the antimicrobial activities, thus dialing out the hERG inhibition or cardiotox liabilities, but improving the drug-like properties in terms of the target and antibacterial activities. So summarizing these findings, what we do at Bugworks is, once we actually identify the therapy area, we would like to look at targets in terms of a multi-target approach, so that the activity is not against a single target, more than one closely associated target, which helps you to gain that much potency against the bacterium, as well as the opportunity to generate resistance is also reduced, because you need to have mutations on dual or multiple targets. Then the lead optimization is done through multiple methodologies, including structural biology, medicinal chemistry, and also looking at various in vitro, ex vivo, and in vivo properties, wherein we look at efficacy. And in all these things, the technological advances are clearly also looked at. So we have a iterative cycle of looking at potency, the pharmacokinetics, the safety, CMC, and efficacy, in the process actually progressing the molecule from a hit to lead to product development. Another very important feature that one needs to keep in mind is it's never possible that your first front runner would turn out to be the best molecule. So it's important that you create a pipeline of products with diversification potential, so that in the future, in case of any property that needs to be tweaked, you would be able to do that on your backups. So apart from the front runner, it is important to have a pipeline of products created, so that you will be able to overcome any other property that you do not want in your front runner. So finally, my last slide, we set out to have a dual target, where we thought we will kill two birds with one stone, where we will inhibit the gyrase and topoisomerase. But in the process, we had to also look at the efflux and permeability liabilities, the hERG inhibition, and also ensure that there is a novel mechanism there. So it is very obvious that throughout this challenging journey, it was not two birds, but we had to kill four more birds in the case of this molecule, to progress it from a hit lead to finally a clinical candidate. Currently, as Dr. Taslim had mentioned, this molecule is in phase 1. We have just completed the single ascending dose, and the multiple ascending dose trial cohorts will begin very soon. And we also are actually looking at, other than intravenous, also looking at oral formulation, so that you have the flexibility of administering either as an intravenous or an oral product, as a standalone or as a step down option. And being a broad spectrum molecule, we feel we will be able to address the needs of patients with multiple indications, be it urinary tract infections, intra-abdominal infections, biothreat, cystic fibrosis, or pneumonia, and so on and so forth. Thank you so much. And this is the fantastic team that we have at Bugworks. Thank you. Thank you so much.
Thank you, Dr. Vasanti, so much for that. The new drug, the BWC0977. I think very, very interesting, and especially clinicians like us, it's very interesting that where we have, pushed against the wall, we don't have anything to treat this patient. Possibly this molecule would be a real game changer, as you mentioned in your. We really look forward to see this molecule. I just have one question: is it an antibiotic, or what? Is it a combination of something, or is it a molecule independent of its own?
It's a single molecule. It's a small molecule. It's a single molecule which is having broad spectrum activity.
And it has shown good results in the phase 1 trial.
Yes, the single ascending dose has been completed, and it was well tolerated. The pharmacokinetics were as expected. The multiple ascending dose trial is just beginning. We ran into an issue of injection site issues, reaction sometime back. So we had to rework the formulation. So early next year, the multiple ascending dose cohorts will start, because now we have a new formulation to begin with.
It looks very interesting, Dr. Vasanti, and I think India looking forward towards Bugworks for having these new molecules.
Thank you. Thank you so much. Thank you.
Please stay with us for more questions to come.
Thank
I
you.
hand it over to Dr. Kapoor sir for taking it forward.
Thank you, Dr. Vasant. Thank you, Dr. Vasanthi, for a wonderful talk and amazing work Bhagwas does. Next speaker is Dr. Christina Ospier from ICAS, a Denmark-based international organization working in the field of antimicrobial resistance. And Dr. Christina Obser from ICAS, and ICAS was established from the leadership of legendary Professor Henrik Wegener, my good old friend who transformed veterinary antibiotics in Europe and led the foundation for the worldwide change in the veterinary antibiotic stewardship. And we are proud to say that Dr. Sujit Chandy, he is Executive Director of ICAS and he is from CMC Vellore. And we thank you for helping us to connect with Dr. Christina. And Dr. Christina Ospier is a global authority in veterinary antibiotic governance. Welcome, Dr. Christina. Over to you.
Thank you so much, and thanks a lot for the introduction. I hope that you hear me okay, and I will see if I can start sharing my presentation as well, and get it in presentation mode. So do you see my presentation now in presentation mode? Okay,
yes.
Wonderful. So thanks a lot for the introduction and thanks a lot for inviting me to present here. As mentioned, my name is Christina Ospier. I'm the Director of Science at ICARS, the International Center for Antimicrobial Resistance Solutions, where I have a background in veterinary sciences, though. I did my PhD in One Health, and I worked my entire career with a focus on supporting low and middle income countries and establishing better mechanisms for controlling key animal diseases, zoonotic diseases, antimicrobial resistance, with a focus on animals, but also looking at the implications for human and animal and environment sectors. So I will just give you a glimpse of what has happened over the past 100 years in terms of antimicrobial use in animals, where we are at today and where we need to go in the future to enable a reduction in antimicrobial use and to mitigate AMR. It will not be as molecular as some of the previous speakers have been. The focus of ICARS is very much testing context-specific solutions in collaboration with low and middle-income countries. So I will give you some examples of that, and also some glimpses of what has been done in Europe in terms of reducing antimicrobial use. So moving, going 100 years back, and giving them some historic perspectives on animal production and on antimicrobial use. So if we look 100 years ago, and also in very many low and middle income countries today, livestock were mainly raised in grazing and scavenging systems, not in the closed-in systems, but in larger areas. But in the 1920s, the US, they started producing particularly chickens in much more intensive systems. So reducing less land for the production of meat and to be able to feed a growing population and to able to meet the increasing demand for meat. In the 1940s, when antibiotics were developed, they also there was testing starting to use antimicrobials in animals. So Gramycin and penicillins were tested against mastitis in the 1940s in the US and in the Europe, and were then expanded more and more in animal farming. And after the Second World War, there was a really booming post-war demand for meat, which pushed the meat producers to increase their production very quickly. Also, this connected the pharmaceutical companies and the feed producers for animal farming, and medicated feed became more and more popular in animal production. So while the animal production was going more and more intensive with a higher density of animals, but with poor husbandry practices and biosecurity in place, it was discovered that you could actually raise animals by feeding them with low doses of antibiotics and maintain very good health. And antibiotics were cheap. So it was an easy way of enabling a high productivity with a low cost and without having to invest in high-level husbandry practices and in better biosecurity. So between 1950s and 1970s, the US non-medical antibiotic use increased enormously from 110 tons to 3,310 tons in 1958. And at that time also, it was estimated that the British pigs, that 50% of the British pigs were actually fed antibiotics on a very regular basis. As I mentioned, it was cheap and it was easy to feed the animals with medicated feed. And also this cheap and reliable protein was very much prioritized as compared to strong antimicrobial stewardship, even though Alexander Fleming already, when he discovered penicillin and when he got the Nobel Prize flagged for resistance levels, I think the growing demand for meat was very much driving a continued antibiotic use in animal farming. Also, the global consumption of meat increased rapidly. So if we look at comparing the data from the 1960s to 2014, it almost doubled the global per capita meat consumption. And you see that in the graph here to the right for the different species, how the meat production was increased in the different continents. You see that Asia is standing for a lot of the growth in meat consumption, but also other continents in the world. And if we look at the past 20 years, poultry has been the fastest growing meat in terms of production levels, but also aquaculture stands for a very rapid increase when it comes to meet the global demand for animal sourced foods. We see also a growing demand for milk, and aquaculture, poultry, and milk production depends very much on access to good antimicrobials, and antimicrobials are commonly used in these production systems as growth promoters to prevent infections. And as this global demand for meat will continue to grow, there are projections that we will also have a continued increase in antibiotic use. And I will show you in the next slide some data on that. When TCO and Thomas van Buckel and the team, when they have been looking at antimicrobial consumption and globally, they found that comparing the 2017 levels with predicted 2030 levels, there is an estimate that it will be more than 11% increase in antimicrobial use. You see here the dark red cycles. They correspond to the amount of antimicrobials that were used in food animal production in 2017. And the outer blue ring corresponds to the projected increase in antimicrobial consumption until 2030. You will notice here that some countries, it's the country level data, that some countries are projected to have a higher increase in antimicrobial consumption. You see, for example, Brazil being highly represented here, South Africa, also Russia, China, and some countries in Western Africa. We also already know that this antimicrobial use is closely linked to increasing antimicrobial resistance. So the second figure is showing the antibiotic resistance rates in food-producing animals compared to antibiotic consumption in food-producing animals. And there are close associations with higher resistance rates in countries that and regions that are having higher antimicrobial use. What is interesting, though, is that there is also a bidirectional association here. So animal antibiotic consumption is positively linked also to resistance in critical priority human pathogens. For example, in carbapenem-resistant Acinetobacter, third generation cephalosporins, and in oxacillin-resistant Staphylococcus aureus. And interestingly, the same study also found that antimicrobial use and consumption in humans were linked to or associated with higher resistance rates in animals. So there is some evidence now showing that the antimicrobial use in animals and in humans are actually influencing the resistance rates in the other sectors. But we shouldn't give up here, but because there are opportunities to change the current practices. So most countries in the world, they report in the ANIMU system, so the World Organization for Animal Health, their antimicrobial sales, the import, and some countries also report the use. So positive trends from the last 9th report that the WOHA released showed that there was actually a declining trend, that antimicrobial use is declining by it was reduced by 5% when comparing the antimicrobial use data 2020 with the 2022 data. And this was true for most continents in the world. You see here, for example, for Africa, there was a quite remarkable reduction in antimicrobial use. There was also a reduction in Asia and the Pacific, the Americas, and in Europe. So the only continent that reported a small increase was actually the Middle East in the latest report to WOHA. Also, we look at the EU. There has been a steady decrease in the sales of veterinary antimicrobials for food-producing animals. You see here the graph to the right showing the decrease where it fell by 28% in 2022 as compared to 2018 data. So what have countries then done in the EU to enable a continuous decrease in antimicrobial use in food-producing animals? I will now give you some examples. We first go to Denmark. So Denmark was one of the first countries in the world that banned growth promoters for animals. And Denmark also introduced a yellow card system in 2013. This yellow card system actually, what it did was that it targeted the farms, the pig farms, with the highest antimicrobial use. So by getting the antimicrobial use data on the farms and by targeting the farms with the highest antimicrobial use and giving them a yellow card, and supporting those farms in really reducing the antimicrobial use, Denmark could reduce antimicrobial use in pig farming enormously. Since these measures were introduced, antimicrobial use in pigs have been reduced by 27% in the Danish pig production. But in addition to applying this yellow card system, where they also in Denmark now make antimicrobial use data on farms accessible by all farms. So you can actually see how much antimicrobial use your neighbor farmer is using, which is further kind of incentivizing farmers to really reduce their antimicrobial use. In addition to this yellow card system targeting the highest antimicrobial use farms, Denmark also implemented a tax system where there was more tax on some critical antibiotics, such as fluoroquinolones and colistines, and third and fourth generation cephalosporins. Another example is the Netherlands. So the Netherlands used a combination of compulsory and voluntary actions to really reduce antimicrobial use in farm animals and managed to reduce antimicrobial use by 56% between 2007 and 2012. And it was a combination of different actions, but one part was that they tied a veterinarian to each farm. So it was a one-to-one system where each farm had to have a veterinarian tied attached to the farm who was providing specific and targeted advice for antimicrobial use within that farm. Moving over to Sweden, in the north of Europe, Sweden was actually the first country in the world who banned growth promoters in animal farming. It was done already in 1986, compared to then the EU who banned it in 2006. Also, this was enabled by a collaboration between the industry, the authorities, and academia, where academia was providing evidence to authorities and what could be done in terms of reducing antimicrobial use, and where much of this ban was actually pushed by the industry because of the consumer pressure, that the consumers in Sweden started to get worried that there was so much antimicrobial use in the animal farming system. In addition to that, antimicrobials can only be prescribed by qualified veterinarians, and you can only buy it you can only buy antibiotics if you have a prescription by a qualified veterinarian. And veterinarians were also permitted from making any profit from selling antibiotics, since all antibiotics are sold by the pharmacists. So there has been a combination of this collaboration, the tripartite collaboration between the industry, between authorities, and between academia, and ensuring that the regulations on antibiotics, that those are in line with what the industry can actually apply. And the theme has very much been that healthy animals don't need antibiotics. So working a lot with disease prevention practices, and thereby enabling really a reduction in antimicrobial use. You might think then that, okay, this is what you can do in a high income country, but what can actually be applied in a low and middle income country? That's where we come in ICARS. We are an independent self-governed organization. We were established in 2019, first starting under the Danish government before we became independent. The focus of ICARS work is to support low and middle income countries to really test solutions to tackle antimicrobial resistance across the human, animal, and environment sectors, and also some cross-sectorial work in this country. What is key is that these solutions, they are based on the country priorities. So it's country determined what solutions are tested in countries. ICARS is co-developing projects in collaboration with the countries in making sure that the projects are generating strong evidence on what works in that specific context in terms of reducing antimicrobial use and mitigating antimicrobial resistance. So we provide both technical input and funding, working with intervention and implementation research framework, and ensuring that there's a plan in how the solutions will be scaled. Sorry, is there a question? I think. Okay, thank you. Shall I continue?
Continue. Some interference. That's
okay,
all.
Just let me know. So we are currently supporting 60 projects in 28 different countries and territories, spanning across human, animal, and environment sectors, working with more than 170 partners. And I will give you a few examples from projects. We're starting going to Colombia. So in Colombia, we've been supporting a project since 2022 on reducing antimicrobial use and diarrhea incidents in pig production through improved access to colostrum, raw milk, and also better use of vaccines. The aim of the project from start was to reduce antimicrobial use by 20% in piglets and through a reduction in diarrhea incidents in the piglets. So it was a before and after setup where it was implemented on 5 different farms, where these farms were followed for 6 months in terms of their normal practices, and then the intervention, that combined colostrum vaccine intervention, was implemented over almost 1 year in the same farms, where you could compare the farm data with itself. So collecting information on antimicrobial use, but also economic data on productivity and so on. This project was implemented, I should say, by Pork Colombia, so the largest pork association in Colombia, in very close collaboration with the Ministry of Agriculture in Colombia, but with the funding from ICORS. What was very interesting here that they exceeded the target. So by applying these good targeted vaccines for farms, guided by the actual prevalence of pathogens on the farms, and improving the access to colostrum by the piglets by rotational suckling, ensuring good IgG levels in the colostrum, the diarrhea could be reduced by half. So there was a 50% reduction in diarrhea incidence in the intervention group as compared to the control group. What is even more interesting is that antimicrobial use in terms of medicated feeds could be totally stopped on the intervention farms, allowing a 91% reduction in antimicrobial use in the intervention group as compared to the control group. So there was a reduction, and the most of this was because of the stopped medicated feed, but also that the number of treatments required for the piglets was dropped. And that enabled the project to meet this remarkable 91% reduction. And despite that, we had this stop of medicated feed. There was a 45% higher growth rate of the piglets. The project is now looking at the economic data. But what's interesting was with these small interventions, antimicrobial use could be reduced, but with even increasing growth rates of the piglets. I will also give an example from Vietnam. We were working in striped catfish in tilapia farming, just to give an example from our aquaculture production project. So the project is focusing on improving morbidity and mortality from bacterial diseases with a focus on bacillus necrosis in Pangasius and motile Aromonas septicemia and Streptococcus agalactia. And using vaccines that are already available but are not widely applied, and seeing what are the barriers and enablers for a stronger uptake of these vaccines in Pangasius and in tilapia, on improving the farming practices and how the tilapia is produced. Interesting here, in the farms that applied the vaccine, so the vaccinated striped catfish had a lower feed conversion rate. They managed to reduce the mortality as compared to the non-vaccinated fish. And the economic data is now being generated, but the reduction in mortality and the feed conversion rate that seems to exceed highly the cost of the vaccines on these farms, and really managed to reduce antimicrobial use, showing that it is possible in different types of animal production system in low and middle income countries to reduce antimicrobial use, to mitigate AMR, but with maintained productivity and with maintained economic profitability, which is critical to convince farmers to apply good mitigation measures. So this is the final slide. In the UNGA political declaration for 2024, there is a target of reducing AMR-related deaths in human by 10%, and also to decrease antibiotic use within the agri-food sector. We need, and the UNGA also calls for more incentives and more research and development on what actually works in terms of reducing antimicrobial use in animal farming. And we need more of these good examples, particularly from low and middle income countries where the highest burden of AMR is projected. But countries need to work in collaboration. And then we need to have stronger collaboration between researchers, the industry, and decision makers. We need more of this specific, context-specific evidence of what works in the specific local settings. and also to be able to show that you can produce animals with lower antibiotic use, but with maintained profitability. So thanks a lot, and over to you as organizers, and happy to take questions.
Thank you, Christina Ospia, for the excellent talk and summary of what's happening in the world of veterinary antibiotic usage. We will have a discussion, panel discussion of all the speakers after the next talk. And I'm sure you will have plenty of questions to answer. And next we have Dr. Jnana Soundari and Dr. Chaitra. Dr. Jnana?
Yes, sir. Can you hear me?
Okay. Oh, yes, yes. Now, we have a session called, they called it crazy, we called it science, the colistin resistance journey. In 2019, India banned the growth promotional usage of colistin. It was the result of work of so many stakeholders, experts, organizations, media. I remember at that time, I was one of the technical advisory committee members for the national policy. And as the coordinator of Chennai Declaration, we provided the evidence to the Indian government why colistin as a growth promotional agent should be banned. Yes, we published a paper, the first paper from the country, on the presence of colistin-resistant bacteria in Indian food samples, and we delineated the molecular mechanisms. I was the lead author and did some political and policy work to get the ban done, but we couldn't have done that work without the scientific support of two young, amazing scientists. Both were postdoctoral, doing their PhDs at that time. And one is Dr. Jnana Sundari Palani, and other one is Dr. Chaitra Sangar. Dr. Jnana Sundari, Dr. Jnana and Dr. Chaitra will tell us the excellent scientific molecular work they did on colistin resistance, the first paper from India. Dr. Jnana, over to you.
Thank you so much, sir, for inviting for a talk. It was always good to see you. And it is our favorite topic to discuss. So I'll share some of the slides. So as Gafur sir said, this is the first study which we have done. So far for the last 1.5 hours, you would have been heard a serious discussion on
No, we can't see the slides. We can't see the slides. You can share click in the arrow below on the middle. Yes.
Yes.
Now it's coming. Yeah.
So for the last 1.5 hours, we are having a serious discussion on various topics. Let us take a small breath. Yes, this
Vasan,
is.
can you see the slide?
I can see the
Vasan, can you see the slide?
No, sir. No, sir.
No, we can't see the slide. Jnana.
Not it, not it.
Just a minute, sir. Just see.
Yes, we can see your slides. Yes.
Yes, sir, can you see now?
Yes, yes.
Okay.
Yes, so what we are seeing now is vegetable, chicken and fish sample. Yes, we also have antibiotic resistance in these samples also. Just 6 years after this study, we are having a discussion now. These samples are having colistin resistance, which is extensively, this colistin is something which we use for last resort antibiotic. But before we collected, this study was not started as the major part to look into. It's a part of the study where we wanted to screen the stool samples for carbapenem and colistin resistance. I remembered once we had a discussion with Kapoor sir. He said, why can't we process the samples to see whether we were able to see any colistin resistant or not? At that time, we didn't have any point that we will be able to isolate this resistant bacteria. There is a lot of questions which we had at that moment. The first thing, how are we going to isolate? Because during that, during 2015 to 18 time, we don't have any media to isolate this bacteria from the, even not from the usual source like clinical sample. But we have a huge number of food source, but we don't know how we are going to enrich the bacteria, what kind of media we are going to use, or what concentration of colistin will be helpful for us to pick up. But slowly, when we collected and we processed some few number of samples, we found that, yeah, it is working. So almost some 110 food samples we collected during October to November 2017. It was chicken, fish, mutton, vegetables, and fruits. So as it's a part of we just mince the sample and then we inoculated in a plate called eosin methylene agar, where you add different concentration of colistin, then pick up all the colonies from that and see whether it is resistant to the drug or not. So what you're seeing now is the plate, eosin methylene blue agar plate, where you see a E. coli which is showing metallic sheen colonies, and whereas you see the other Klebsiella like colonies, which like mucoid kind of thing. So the other two pictures are because of inoculum effect. These are very difficult to isolate because the antibiotic plate which we normally use is affected by the term called as inoculum effect. We could isolate almost some 71 isolates, where out of 110 samples which we tested, almost 51 samples where 46% of samples were colistin resistance. More than one bacteria was isolated from almost all the samples which came positive. It was 12 chicken, 11 fish, 3 mutton sample, 23 vegetable, and 2 fruit samples which was found to be positive. It is the majority of them were Klebsiella pneumoniae. The other organisms are like pseudo acinito, which we normally used to see in the clinical setup also. But the prominent one, which the Klebsiella isolates, had very different kind of MIC compared to the other organisms. Like for example, the E. coli has only very less MIC, didn't even cross 8 microgram per ml. But the other organisms like Klebsiella, Citrobacter, Enterobacter, they have reached even 128 microgram per ml, which we normally see in the clinical setup. And all the isolates which we got from these samples were sensitive to most of the drugs tested, and very few of them were resistant to cefotaxime, ceftazidime, which we normally see in only the stool kind of samples, not in the patient population. In a patient, in a clinical setup, all the drugs will be resistant, including imipenem, carbapenem. They will be a multi-drug resistant sample. But in these food sample, in spite of colistin resistant, all the other antibiotics, most of them were sensitive. The good thing is they are not resistant to carbapenems. It's a very complex slide where there are so many genes which is involved in colistin resistance, but we always speak about MCR, MGRB. Why are they important? Like we all know MCR, which is present in a plasmid, is it can spread from one bacteria to the other. But the other one is usually present in the chromosome, which we normally think it is present in a chromosome. Why it is going to spread? Like in the clinical specimens, when we got to know that this mechanism is completely different from the other usual mode of colistin resistance. We almost got 29 colistin resistant Klebsiella isolates, which we looked for. First, we looked for MCR1, where we found almost only E. coli samples were resistant, but remaining all the other Klebsiellas, Enterobacter, Citrobacter, they are not resistant. They don't carry the normal MCR1. During 2016, the whole world is fascinated with MCR1. Everybody looked for MCR1. That is the one which is causing colistin resistance. But when we analyzed the food samples, only E. coli showed resistant to MCR. But the other Klebsiella isolates, they don't have MCR. So we just thought, what is the other mechanism which can cause this colistin resistance? And this kind of the one which you are seeing here is this is MGRB gene, where it gets inactivated by a small fragment of DNA called insertion sequence. This insertion sequence, this kind of structure you will see in a clinical specimen like blood sample. But when we were able to see this in this food samples, we thought, yes, this is what the mechanism which we normally found in the clinical also can be present in the food sample. So basically, this is a small fragment of DNA which can go and sit inside a gene like MGRB, and they cause a resistance. And we could also see in the other kind of samples like chicken, fish, where it is all mediated by a insertion sequence, which is a family of IS5 or IS1 kind of insertions. Usually this red color, it's a normal MGRB. When this fragment of DNA comes inside and sit near the MGRB, there is either it will sit in the promoter region, so the gene gets inactivated, or it will sit at the either ends of the gene, near the MGRB gene, so it becomes functionally it cannot proceed. Or it can go inside the MGRB and inactivates the gene. But usually this whole kind of mechanism is well established in the clinical setup, not in the samples like chicken, fish, or vegetable. Out of 29 Klebsiella pneumonia which we isolated in these samples, almost 6 samples have this insertional inactivation, which is the reason for causing colistin resistance. And we also could find there is a complete deletion or the wild type, which we normally found in the other clinical specimens also. So this kind of mechanism, when a small fragment of DNA, which is sitting everywhere, is it can go and insert in every segment of the DNA. It can move from one bacteria to the other, or it can move in between the chromosome of the Klebsiella pneumoniae. And even we could see in the other different samples like vegetable, fish, poultry, chicken, mutton, sources, where it is also similar to the MICs which we normally found in the clinical specimen.
Yes, Jnana?
She's lost network probably.
Jnana, you got network problem? Jnana? Okay.
Sir, I need permission to share the screen because I think she's
Oh,
still
you can try.
sharing.
Oh, yeah. Try. Jnana? Jnana has a network problem.
Okay.
Yeah. I think Jnana is still sharing. Yeah, Chaitra started sharing. Okay. Next speaker is Dr. Chaitra Shankar. Dr. Chaitra Shankar at that time in 2018-19 was doing her PhD at Christian Medical College, Vellore, under Dr. Balaji Virajagavan. Initially, Dr. Jnana, who was doing her PhD at Madras University and later as a research assistant at Apollo Hospital, found out some remarkable findings first time in the country. And that is the presence of colistin-resistant bacteria in food samples in the country. Not only that, at that time, yes, even the MCR was new, but when she did that work, she found out something else. Because in clinical practice, EMCR producing colistin resistance is rare. Most of them are chromosomal mutations like MGR. And nobody in the world have done any work on the chromosomal mutations presence in the food bacteria. And we know that MCR spreads from food to the human bacteria, but nobody had looked into whether the chromosomal mutations can spread from food bacteria to the human bacteria, and that is much more important for the human colistin resistance than the MCR. And Jnana did found some insertion sequences which are simply jumping genes that can help the spread of this resistance, colistin chromosomal resistance from food to the human. That was the first time discovery in the world that it can spread from the food to humans through insertion sequences. And then definitely we wanted to reconfirm and do additional work from another laboratory. And CMC Vellore, Dr. Balaji and Chaitra was so willing, so keen to come forward to help the cause, because we had a bigger cause of getting the colistin usage as a growth promotional in the country banned. And Dr. Chaitra did an amazing work along with Dr. Jnana and Dr. Balaji. And thanks, Chaitra. Please help explain to the audience what amazing work you did at CMC Vellore at that time.
So thank you so much for giving me this opportunity to share what we worked on together. I hope you can see the slides.
Yes.
Okay. So once Gnana confirmed a lot of the findings that she just presented, we received the samples for more of like molecular confirmation, and also at that point we had ion torrent, which was one of the whole genome sequencers that was used in the beginning. So we went on to do whole genome sequencing to confirm whatever we found, or rather what Gnana found. So some things that I just want to highlight is that when it comes to molecular mechanisms of colistin, right, like as Dr. Gnana already mentioned, MGRB disruption by insertion sequences is one of the very common mechanisms that we see in a lot of clinical isolates. But again, I want to specify that this is specific to Klebsiella pneumoniae. Like you cannot extend this to other pathogens like E. coli or Acinetobacter, because when it comes to the colistin resistance genes in case of different bacteria, it's got more to do with the bacterial outer membrane, like the lipopolysaccharide region of the bacteria interacting with the colistin. So each bacteria has its, or each genus, I would say, has its own set of genes that help in the lipopolysaccharide synthesis. So the genes involved are very different from like Klebsiella to E. coli. So when we look at MGRB, it's specific for Klebsiella pneumoniae and it's not for E. coli or any other pathogens. And since in this study, Kleb lacked a lot of, I mean, it did not have any plasmid-mediated resistance. So MGRB disruption was one of the commonly reported mechanisms, and that was explored. So I think Nana already mentioned that there are several ways in which this MGRB gene can be disrupted. So basically what happens is, in which case, there is a change in the charge of the outer membrane, and hence there is not enough binding with colistin. And when it comes to MCR acquisition, it was just in one isolate of E. coli that we were able to identify. So we went on and performed whole genome sequencing, because the main interest was to see whether it is one of the circulating E. coli strains, which is also seen in clinical specimens, but it seemed to be quite different. So I mean, this is the MCR1 gene that was identified through whole genome sequencing. But the challenge we faced was that we used the Ion Torrent platform at that point. And in Ion Torrent, what happens is that you get small fragments of the whole genome. So we do not get like a complete plasmid or the complete sequence of the chromosome. So unfortunately, we could not establish the whole structure of the plasmid that carried this gene, but we were still able to confirm that yes, this E. coli does have MCR1, and this particular strain was not something that was commonly circulating in the clinical specimens. So we do know that it's different in the animals, and there is a difference in the niche for all of these E. coli strains. However, this E. coli isolate did have two different plasmids, so we know MCR could be in one of those. And like I said, MGRB is more for CLEB, and then MCR1, although is seen among different species, like in our study, we found it only among E. coli isolates. So coming to some of the challenges when it comes to colistin resistance. So as I said, there are like chromosomal mutations, and there are several housekeeping genes involved, which, as I mentioned, is basically the genes involved in the lipopolysaccharide production. And these are like species specific. So a lot of these genes can have mutations, uh, which can either be truncations or disruptions by insertion sequences. So these tend to generally show up as having higher MIC to colistin when they are tested in the lab. And most often, this is predisposed by colistin usage because it's easy for the bacteria to have chromosomal mutations and adapt to a stressful situation than acquire a plasmid and take on the cost of having to bear a plasmid. And the other thing I want to highlight here is that a lot of insertion sequences are very common in genomes such as Klebsiella pneumoniae, which normally tend to have insertion sequences just present in either the chromosome or the plasmid. And if we look at a lot of reports like coming up in the recent years, and all because of increase in the number of whole genome sequences, we know that a lot of times when there is antibiotic selection pressure, these insertion sequences do play a role in jumping and disrupting housekeeping genes such as like, you know, the MGRB, or it can also be sometimes like the efflux pumps that we earlier heard about. So all these genes can be challenged by like insertion sequences. So that is something which is very tricky. So all these also may give us challenges in identification, because when we are looking for epidemiological reasons, when we are looking at a specific chromosomal mutation, excuse me, it's almost impossible to come to a conclusion that it's this particular gene in this species, because without whole genome sequencing, we have no idea about all the genes that are involved and which gene can have mutation, because several of them, like I said, can influence the MIC. But when it comes to plasmid mediated MCR1 gene, so its identification is more easier, because at least in PCR, you know, if you're looking for this MCR1 gene, whether it's present or absent. But then again, there are several variants reported now. And another challenge of any plasmid mediated resistance is that they can jump from one plasmid to the other and also spread from one species to the other. They can also spread across niches, like the sample that we found, E. coli, which was basically from poultry. If when it comes in contact with, like, say, the person handling that particular sample or so, the bacteria as well as the plasmid can spread to the host, to different species as well, because it's mediated by plasmids. And another thing is with MCR, most often there tend to be lower MICs when, yeah.
Dr. Chaitra, as a housekeeping one,
Okay.
we are running late because previous sessions
Okay.
got late. We may have to hurry.
Okay, yeah, I'm done. So MCR tends to give us low MICs. So I just want to mention that these are some of the challenges, and I think these were explored by whole genome sequencing, and we were also able to compare how similar the food samples were with respect to the clinical samples, and a lot of these MGRB mutations that we saw are very commonly reported in clinical samples as well. Thank you.
Thank you, Dr. Chaitra. It was an amazing work done by these two young scientists. For the first time, working on the food Klebsiella, identifying MGRB mutations, insertion sequencing, defining the pathway, how it can spread to the human bacteria. That was the first time in the world. The whole world was at that time obsessed with MCR genes. And in the clinical practice, the colistin-resistant clinical isolates, very rarely you find MCR. They are all MGR and chromosomal mutations. And these are the two young scientists for the first time worked and delineated that particular food to human transmission pathway. Thank you, Dr. Jnana and Dr. Chaitra. I'm sure some other day we'll have a detailed discussion on this particular topic because we want to hear details about the amazing work you did. Dr. Ashwini, next talk.
Yes, sir. Yeah. So moving on to the next session, that's by Dr. Soumya Sridharan. So Dr. Soumya Sridharan is an infectious disease consultant from Chennai, and she is known for her empathy, creative thinking, and strong patient that fuel AMR. Over to you, Dr. Soumya.
Thank you so much, ma'am. Thank you, Gafur sir and the AMR Trust for giving me this opportunity to talk. After listening to all our stalwarts on so many breakthroughs that have happened in AMR, I have just simple things, maybe the human touch, the empathy that AI can't do at this time, that maybe passion, that fuels our passion for working and making AMR a success, at least in curbing the resistance to at least as much as possible as humans. So I'm just going to have a few case scenarios that I came across that set me thinking as to what we can do from our side for reducing AMR to an extent. So first one is actually this patient is currently admitted in hospital and I have no clue what I'm going to do tomorrow. This is a 56-year-old male who was a CKD patient who had CA rectum from 5 years before with ureteric stricture. So that set off the probably the renal dysfunction for him, and he continued to have obstructive uropathy, which made the vicious cycle of urinary tract infections start for this person. And he came to us with a left-sided pyelonephritis with an ESBL bacteremia. And he was doing well. We had CT showed a collection and we drained the collection with the pigtail. Then he again became hypotensive and he was shifted to the ICU. Urine cultures this time showed a carbapenem-resistant Klebsiella and ESBL E. coli. Again, we had to escalate antibiotics for him and he was better. So we treated him for 3 weeks with antibiotics. And repeat imaging actually was better and hence the pigtail was removed. But on the last day when he came to the OPD for follow-up, he again had one spike of fever, and this time it was an enterococcus. So as you can see, he has started to select out organisms for which he was on antibiotics, and now this was a enterococcus faecium. Again, got vancomycin. Again, became shocky. We had to give him a shot of colistin given the background of carbapenem-resistant Klebsiella. And we stopped the antibiotics. All that he could probably get now was a nephrectomy because this was probably a non-functional kidney. But the family was completely drained out. There was no money for surgery. They were not willing for staying without a kidney. So again, he has come again with shock and I really don't know what to do. To cut a long story short, this was a patient with a complicated UTI and now there are no antibiotic options. Probably it's going to be colistin for him, and if he is going to get a colistin resistant bug, God knows what. We are all going to pray for this person. The next case again is a similar lady, is a 62-year-old lady. Actually, this person is a cancer survivor. So she had ovarian carcinoma and she was operated. Again, came to us with a pyelonephritis. For some reason, all pyelonephritis and urinary tract patients love me, and they come with an abscess, and she was drained, and this was a CR Klebsiella again, and she received ceftazidime avibactam. We thought she again came back after adequate treatment for 3 weeks with repeated infection. So we thought it was the DJ stent that was infected this time, and we removed it. And again, the culture showed the same bug. This time we gave her colistin, but again, despite colistin, she came back with repeated infection. This time there was no stent, and it was a colistin resistant bug. So we had to use cefepime zidibactam for her under compassionate use for this patient. So this was an elderly lady. Actually, her cancer-wise, she is cancer-free and is not on any medications. Unfortunately, she has a CCRE sepsis for which we had to use cefepime zidibactam. She's like a ticking time bomb now, and if there's another bout of infection, at least by next year, we should have this molecule to treat her. This last case, third person, is a 33-year-old male. This I wanted to bring up for saying that it is not just antibiotics that have become resistant. Even antifungals, we are losing out right now. This patient had a CSF diversionary procedure, and his CSF actually grew carbapenem resistant Acinetobacter and Candida auris. So these are the images for of the patient who had a VP shunt infection actually. And as you can see, he came into with us with a frank meningitis. We did, and Candida auris had all high MICs to most of the drugs that were there. So we did not have much of an option, and his GCS was poor. So he underwent a VP shunt removal, and we placed an EVD. Meanwhile, we also ordered an antifungal susceptibility for this patient by broth microdilution. Empirically, the only problem is that Vitek showed us echinocandins were sensitive. But again, we do not know. This is a sanctuary site, a CNS infection where I couldn't use echinocandins for him. So the literature regarding intrathecal caspofungin again is very scarce, and we ended up giving him intrathecal caspofungin along with amphotericin deoxycholate. After the broth microdilution, this was the culture report of the patient. And after the broth microdilution was back, we chose to give him only amphotericin and flucytosine. By God's grace, this patient improved, and he did have residual neurological deficits. So, but he improved clinically, and he's off any medicines right now. This again is like a chicken or egg story. Which came first? Did the repeated antibiotic and antifungal exposure cause this Candida auris meningitis, or did he? It's just like a chicken and egg story. We do not know if the resistance came first, or because of the resistance, we end up using high-end antibiotics and antifungals. So we actually published this. But the other side of all these three patients, if we see the first case, he's a sole breadwinner. He's a farmer, and he has 3 college-going kids, 2 daughters and 1 son. He doesn't have any insurance, and he had to pay out of pocket. He almost sold the entire property that he had by the time we advised for a nephrectomy. And he couldn't afford further surgical care nor hospital care, and he couldn't afford the antibiotics. The second one, and every day when the family comes and meets me, I really didn't have anything to tell them as to what went wrong. They kept asking me, what did my dad do wrong, or what did we do wrong? There's no answers to these kinds of patients. And the second one is actually an affluent family. Here, the cost was not an issue, but we did not have any therapeutic options for her. Although she fought off the cancer and she could afford any drug that was possible, at that point, we had very little therapeutic options. The last one is actually a very young, unmarried male. And when the mom asked me, he is alive, but will he get married and will he lead a normal life? I really did not have any answers or solutions to all of those problems. Probably AMR cannot answer all of these, and we do not have much of literature to give more medicines on top of almost everything that they've had. We've exhausted all options that are there, and we really don't know. So AMR is actually real. There is a test every 45 seconds, and the routine procedures, even the surgeries, most of the times when we see smaller hospitals for normal delivery, they is prescribed antibiotics, and the lady goes home with 5 days of antibiotic prescription. So the maximum burden of AMR actually is in the Southeast Asian regions, where you can see the resistance, especially to gram negatives, is very high. And what keeps us fighting AMR every day? Is it because of the patients that we've lost, or the families who trust us and come back again to us for answers, or the belief that we think that innovations can outpace resistance? I really don't know. We fight AMR not just as a science, but for the people. So once we put the science behind and think about the people and their other side, probably we would not prescribe antibiotics as rampantly as we actually do. And we all cannot solve all of the crises of resistance, because it's not abstract, and mostly it is a personal story that we have to see in terms of antimicrobial resistance. These stories are not rare anymore, and every other day we see such stories, and maybe we need some more ideas, some crazy ones that we can start thinking of. As Arvind sir said, I had a few suggestions like him. It was a very nice initiative that actually Kerala has adopted. There is always a lag between the discovery of new antibiotics and antimicrobial classes. As we all listen to so many lectures, that so much of science, development, research, and money goes into developing new classes of agents. And unfortunately, these classes get used up so often, so fast, that we do not have further options to treat our patients. Next, probably there are vaccines. Again, there are so many new innovations that are happening in the field of vaccines that we just listened to. There are precision and targeted therapies like the phage therapy, the CRISPR-Cas system, and the monoclonal antibodies. These are all under research, but by the time we have a solution with these, it must be outdated already. Like how our bugs develop resistance to the antibiotic that are yet to be almost discovered. So microbiome-based interventions are also on the pipeline, where we see probiotics, postbiotics, and fecal microbiota transplantation to restore healthy flora. And AI is here to stay. Probably there is a lot more things that we probably can use AI for in the field of antimicrobial resistance. There are certain natural products, and India which is known for its rich heritage. Neem, turmeric, probably sea sponges. All of these can be looked into, because we have a rich biodiversity that we can probably innovate new products. And again, antivirulence and quorum sensing inhibitors, these are also on the pipeline. These are areas where fast research is happening to reduce the selection pressure for resistance. Finally, it is a One Health and an environmental innovation approach, where we have to reduce antibiotic use in livestock and water surveillance. We could actually implement water surveillance for AMR genes, and it could be an early warning system saying that the AMR in the water surveillance has increased to actually curb or limit the antibiotic use and spread antibiotic awareness amongst hospitals and also the community. Finally, we need administrators, we need policymakers to implement strict stewardship, not only in the Tier 3 hospitals, that is tertiary care hospital, as Sir was saying, in the PHC level. If it has to start in the grassroots level, then we could nudge, we could do audits and feedback, we could see where each hospital stands, and probably incentivize hospitals with a low AMR pattern so that their prescribing patterns come down. Finally, these are a few ideas which I thought of, may not have any sense, but still maybe 20 years down the lane, I can say we can see if this actually made through or not in the field of antimicrobial resistance. Probably we could have predictive AIs, like doctors receiving prompts through AIs. As we all ask our ChatGPT friend, we could probably have an app which can actually help doctors to make decisions on resistance risk, and also prompt them that this is an unapproved combination while prescribing the antibiotic. And also give incentives to the healthcare provider. Probably a good doctor can be incentivized for the least number of prescriptions. Although this universalization may be difficult. A surgeon might prescribe less, a physician might prescribe more. But amongst the surgeons, we can have something sort of an incentive. And again, this is digitalization of antibiotics. We can link the ID card or the Aadhaar card of the patient to the last antibiotic prescription, at least when that happens, we could see the list of antibiotics that the patient has actually received to actually titrate our prescriptions, and the antibiotic return programs, as Sir was saying, to probably give back the unused antibiotics rather than keeping it at home, because it tends to be used by the family members. They give it to their neighbors. All these can be avoided if it is just a prescription-based antibiotic that happens. The number of antibiotic used would also be reduced. Thank you so much for the patient listening. Thank you, sir, for the opportunity.
Thank you, Samya, for the real life cases. All of us struggle with these kind of cases day in and day out, because majority of the time we do deal with these MDR bugs actually. And the crazy ideas were really good, and quite a few of it were actually implementable. So we can think of, especially if Arvind is listening, Kerala is a state which is implementing all this. So probably they can start implementing and we will follow them up. So we're running short of time. We'll move on to a panel discussion. I'll invite Dr. Kapoor sir to take it forward. And before that, if audience have any questions, please put it in our chat box. We'll try and answer them. Over to you, Kapoor sir. Thanks, Soumya.
Thank you, Dr. Soumya. Soumya gave us the front line, on the ground realities. We had amazing discussion by scientists, clinicians, innovators, all crazy ideas that became a reality. You know, the German philosopher Arthur Schopenhauer once told, all the truth passes through 3 stages. First, it is ridiculed, then it is violently opposed, and then accepted as being self-evident. That's what is happening. We have so many questions by panelists. I believe we can discuss for hours. What we will do, because we are running late by 20-25 minutes, we will conduct individual separate webinars by all these speakers. We need detailed discussion by all of them. We will postpone our question and answer. We will try after our webinar on AI and stewardship. After that, if speakers are available, and we can discuss these questions. Let's move on to the panel discussion of AI and AMSP, because human intuition needs a crazy partner. If there is one field today where crazy ideas become reality faster than we can imagine, that is artificial intelligence. What sounded impossible a decade ago, AI predicting AMR trends, detecting outbreaks, supporting stewardship decision, is steadily becoming a reality. But how do we bring bold, unconventional AI ideas into antimicrobial stewardship? We have 6 expert panelists. We have got 3 panelists from APUA, the Alliance for Prudent Use of Antibiotic. Professor Pierre Tatovin is a chair of APUA. He is a professor of infectious diseases from France. We have got Dr. Gabriel Vihara. He's also a APUA board member. He's from Argentina. He's an AMR leader in South America and the world. We have Dr. Johan Scruton. He's an APUA board member. He's an intensivist, infectious disease physician, and stewardship expert from Netherlands. And we have got 3 participants from India, all representing AMR Declaration Trust. We have Dr. Yamuna Devi. She's a trustee of AMR Declaration Trust, microbiologist, infection control expert from Apollo Hospital, India. Dr. Nitin Bansal is an infectious diseases consultant, a trustee of AMR Declaration Trust. He works at Rajiv Gandhi Hospital in Delhi. We have got Dr. Vasant Nagwekar. He's a deputy managing trustee of AMR Trust, a senior infectious disease physician in the country. Let's move over to the panel discussion. The first question is open to all. What are the biggest limitations of human-driven stewardship? Why do we need AI? We are all experts. Why can't we just continue to do stewardship without AI help? Pierre, over to you to begin the discussion.
Thank you very much, Abdul, and thank you to all the speakers of just before you. That was very interesting, and I found very stimulating the talks by Dr. Soumya, for example. What I think is probably the task is so huge in terms of antimicrobial stewardship that we may never be enough staff to be able to resolve the problem of antimicrobial stewardship in every context, in every part of the world, in the big countries, in the big city, but in the rural area as well. So I think that's one of the first reasons why AI may be very helpful to try to improve in the field of antimicrobial stewardship is that it can save time for the doctors, for the nurses, to have more time to save for their patients and to spend less time just to try to identify the best tools for antimicrobial stewardship.
Dr. Nitin?
Yeah, sir, good evening all. I just wanted to go back to the very basic question of what is AI actually. So, can we define what is AI? Is the AI basically data science or interpretation of data, or is AI machine learning? Because if that is what is AI is, then we are already in this era. So, is AI already being used in AMS in some hospitals, or if some expert can give some insight into it?
You're on?
Yes, thank you very much, and thanks for that great question indeed. What are we talking about? I mean, there's so many different synonyms for what we would call AI, right? We have language learning machines, we have machine learning, we have indeed all types of different systems available. And then in the field of infectious disease and microbiology, there's such a diversity of what we could approach with AI. I think at the moment, but I mean, please, others they can contribute if they know more. But that we're still in the beginning phase of actually starting to use these kind of machines in our daily interpretation of on the clinical side, right? I think there's definitely a field out there, and maybe more now at the moment in the scientific space in developing these models. There's a lot going on at this moment, at least in my clinical practice. I don't have a real a close connection to AI models that I use in my daily practice. That doesn't mean that I don't want to. I really would like to, but I'm still quite uncertain in how to use it. And I think that is something that comes across with a lot of clinicians like you all are. How do you now start to use this in a safe way, and how do you now start to make sure that what you are using really leads to better outcomes? So that will be the main questions I'd have.
Thanks, Euron. Gabriel, why do we need AI? We are all experts. Why do we need really AI for antibody stewardship?
Hello, everybody. Hello, nice to see all of the friends. Yeah, but the simple answer, apart of all the things that our friends Sharon and Pierre said, is that we are not all expert, or people that use antimicrobials is not expert. Beside the fact that in our low resource in middle income countries, we are really short ASP team. So probably we will discuss it during this hour, using smart AA and adapt it to our grounds probably can help us. Just to say a little bit of this.
Right. Gabriel told we are all not expert. We have some expertise, but that's not enough. Dr. Yamuna?
So I think the number of infectious disease experts and the number of microbiologists, all the people interested in doing infectious disease, we are not to that extent the way by which we can follow up the stewardship practices. And the data is enormous, what's generated on a day-to-day basis. I think AI is the way forward by which we can have the data which gets standardized and summarized, so that the skillful or the knowledge of the clinicians or the physicians are being utilized only after the data gets analyzed and comes to us, rather than looking into the data and being a data analyst all the time. That work would be taken over by the AI, and the skillful work would be attributed in giving the appropriate follow-up, or in giving the appropriate decision-making skills has to be done by the clinician and not the data analytical part. And that's where I feel AI has a very important role to play in stewardship practice.
Right. Dr. Yamuna stressed a very important point. There is so much of data being generated by the microbiology lab, the antibiogram, and the clinical data, dozens of parameters, and so many things are happening, both the lab and the clinical world. And we few members by the infection control antibody stewardship team won't be able to analyze all this data live, because our analysis, retrospective, and that may not be able to help us to identify red lines or flags live. We need some help to analyze all the massive data. And as Gabriel has mentioned, not all of us are expert. We may have some expertise, but that's not enough. And expertise is so heterogeneous around the world. Vasant, you are a clinician. Do you really need AI for your antibody stewardship work? Please switch on your mic, Vasant. Your mic.
So we are not using AI for tackling AMR in our hospital, but I think that's the way forward. And I think there are a few things which AI would help in the hospital to tackle the AMR. And the first one is to, the antimicrobial stewardship. Because AI-powered decisions will help the clinicians to choose selecting the appropriate antibiotic. That's the first part. Second is, I think AI also by having a good infection control can analyze the data to identify patterns that indicate the risk of increased infection spread which is happening. So I think that is the second part which will.
Yes, Dr. Vasan. So we are all sure that we need AI for antibody stewardship to help us wonderful, intelligent, beautiful humans. We need the digital, emotionless help to do the background work of analyzing the data and giving us the red flag so we can communicate with the people and do the real work in a better way. We need AI as our expert, intelligent assistant. Now, Raksha, you have done some work in AI, in AMSP and infection control. You have got more technical knowledge than we do have. We need explanation of some simple terms. For example, what is rule-based AI? What is machine learning? What is LLM? Or what is ChatGPT, the term? So we need some basic explanation for us and the audience.
Yes, sir. So thank you for the opportunity to share my little experience with the models that I have worked with. So when you speak about rule-based learning, traditionally, the way we have been taught of using antibiotics would be your standard treatment guidelines. You refer guidelines, textbooks, then go by the prescriptions that your seniors or your unit heads would be giving it in your regular OPDs. But when that would be like something you call it as rule-based learning. That's what we have been taught. That's what the guideline says. But when you talk about machine-based learning, you're looking at something which is more real-time, more customized to your setting of work. It could be a tertiary hospital or a primary hospital. Then again, there's a concept of what category of patients are you catering to. So you look at your own data set, look at your own guidelines, use algorithms which are more customized to your center. You could use any machine learning software that is more centric to your care. So what you're doing is you're feeding in data. There's a concept of Ouroboros here, what we have learned from the Egyptian culture. No, it's like a snake feeding its own tail. Garbage in, garbage out. The data that what you feed, what Dr. Yamuna was saying was very right. The structured data, what data we currently have across the globe is not very uniform and structured when you compare LMIC countries or countries abroad. You look at softwares which are already developed in other countries, but in India, if you have to work on these kind of softwares and see, I don't think there's a connect between people who are working on this and the hospitals which are actually prescribing antibiotics. That is one area where I think we can do something and have customized models in our own centers. It's not impossible. It is definitely possible. What Dr. Soumya was telling was very right. It is not very far away. Maybe in another 5 or 10 years, we must be having customized to our own clinics. Maybe Dr. Gafur can have one for himself. Dr. Vasant can have one for himself. So it's not very far off. The idea is not very far-fetched. We need to work on these kind of concepts. And it is not that ChatGPT is the only large language model. These large language models only focus on the data, what we call as tokens. What we feed in, it will give you an output, a structured output. But machine learning softwares look at your antibiogram, the patient cadre that come to your OPD, It looks at models like logistic regression, random forest. We need to learn these data analytics component not to become data analysts, but to understand what models they can make for us. I think this is a gray area and of course a lot of potential for us. And like Dr. Nitin said, what is artificial intelligence? I feel it is adaptive intelligence. There's nothing artificial about it. And we can't beat microbial intelligence. We can't beat it. We can only adapt to it. And we need to be faster than them in mutating, is what I feel technically. We need to mutate our knowledge and it has to be more real time.
Thank you, Raksha. So we have got three reasons why we need AI. First, we don't have that much of expertise. We are
Yes.
dealing with too much of data. We need help.
Yes.
And three, we need to outsmart the bugs that we
True.
don't have the capability. We need AI for that. Three reasons. And Raksha explained, Raksha is an AI expert. So for the sake of the audience, I did some chat GPTing this morning. I learned the exact meaning of the symbol term. Rule-based AI means it's working on defined rule. For example, the creatinine is high, reduce the dose of antibiotic. That's a definite rule where the AI algorithm doesn't have a freedom to go beyond it. While machine learning is entirely different, it has the freedom. It learns from the pre-existing data that it's learning, that we have fed to the algorithm. It can learn pictures, diagram, chart, whatever it gets, it learns and then predicts. It has the ability to predict. It goes far beyond algorithm or predictive rules, the rules defined beyond the rules. It has the freedom to predict. That is machine learning. And what I learned, large language model means mainly text-based input and text-based output. Large language model will help us to analyze large data, text-based data. Well, ChatGPT is a type of large language model. We use images, codes, music, and everything. That's a multi-model. So there's a rule-based algorithm, AI, machine learning, large language model, and then the multi-model, like a ChatGPT. That's what I learned, very simple. Now, yes, Nitin, if Indian hospital want to implement AI for stewardship, but most of our hospitals do not even have a digital database, OP and IP. Still, we don't have digital. Is it possible to install AI, implement AI for AMSP in the absence of electronic medical records?
I mean, sir, there are two ways of looking into it. First is the adequacy of the infrastructure, which will obviously require the digital infrastructure in terms of number of computers per patient or number of computers per healthcare worker, which itself is not defined. In fact, if you look into the any literature, there is no defined literature or standard of care which defines that this number of computers should be there per healthcare worker. So our hospital is currently going through this digitization, and we constantly struggle with the number of computers. The number of computers are far less than the number of healthcare workers. So we have to wait sometimes in line that one consultant writes his or her note, and then the other consultant has to write his or her note. So those things are there. But as far as even with minimum infrastructure, then also we can use AI, but that would be largely rule based AI, algorithm based decision making, like you gave example of high creatinine, reduce the dose, or this patient does not fit into a risk factor for giving CRE cover, so you do not need to add CRE cover, or do not need to add antibiotics. Those kind of simple a questionnaire a clinician can fill in and get the right answer for the correct antibiotics. Those things can be done. But if you want to individualize, like we were discussing, like Dr. Raksha said, individualize into every aspect, we need to then have very high end of infrastructure, digital infrastructure. And second aspect of is the privacy issue. So you can use algorithms, or you can use the mechanisms which are being used in some other private hospitals, large hospitals. Just for example, let's say in Chennai, some hospital is using that. Can I use those machine learning, whatever machine has learned, can I use it in my hospital? That means there will be breach of privacy. So all these aspects needs to be taken care of before we jump into this in the world of using software to make decisions, individual clinical decisions.
Yes, Nitin, as rightly said, it's nice to talk about using AI, but there are a lot of safety implications and the infrastructural issues. Pierre, in France, are you using AI for stewardship?
I was very interested in listening about what AI can be and how it can help us. And I was just thinking, part of my job, I am working in a referral hospital as an infectious disease in France. And part of my work is to give advice to the surgeon because they don't want to be expert in antibiotics. So maybe we got 20 advices per week from orthopedic surgeons to know how to treat bone and joint infection, or 20 advices per week from neurosurgeons. And I think a kind of machine learning should be able to address most of these advices. And probably if they could just listen to 100 or 1000 of the advice that we are giving every week all over the world to the surgeon, maybe we could have like a platform that could resolve most of their problems. It would have been to be controlled by someone who is expert in the field just to check that the advice is appropriate. But it could save a lot of time, I think, if we were supported by AI when we are giving advices to jobs like surgeons or intensive care units or people like that who don't want to get involved in the AMS too much.
Is any of the hospital in France using AI for AMSP?
Yeah, I would like, but as I told you, I'm not really an expert in AI, so I still have to learn on that. I have my own learning process to do about how I can use AI, but I'm convinced that it can help us a lot.
Right. Yes, agree, Pierre. Euron, any
Yes,
of the Dutch hospital using?
Yes, well, just to mention, I think this is something that's coming up, and we see it happening in really small, if you want, places in the hospital where it's sort of protected places. I know that there's some providers who increasingly start to use this. We use Epic as an EMR system in the Netherlands quite a lot, and I can see there's a lot of development there. I think what was mentioned rightfully, that this sort of rule-based decision making on, well, if you say simple algorithms like, okay, your creatinine goes up, you need to change your dose. I think those are evident, and they're already quite far developed in the space of computerized decision making. I would call that sort of the precursor of what we're doing right now. We already have a lot of computerized decision making tools in our prescribing system, right? So if I prescribe a drug, it will say, "Hey, listen, it's better to give this dose because we see the creatinine in your patient is this or that." That I'm not sure if that's actually even AI. It's actually just combining information and then giving you the right advice according to what the guideline would say. What I'm psyched about, and I'm very interested in, and that makes infectious disease and microbiology so interesting, is that the contextualization is so much more diverse and so much more complex than it is for treating hypertension, right? Treating
We lost Euron. Can you hear me all?
Yeah, I can hear you. I also lost Sharon.
Yeah. We hear you very well, Abdul. Yeah, oh, fine.
Gabriel, what do you think? What's happening in Latin America?
Well, this is a problem. This is we have with the Pan American Health Organization been working a lot and performed a checklist for antimicrobial stewardship programs during 22 and 23. For example, you can see that from 1,120 hospitals that were survived, that worked with us during 2022 and 23. We know that, for example, in the checklist, we know that less than 20% of hospital has electronic clinical records, and a short proportion has electronic prescription or interfaces connecting clinical records with microbiological lab results. So we have a short number of terminals of PC or computer available for prescribers. There's not interconnectivity, so we have to work a lot. I have some ideas to share with you later on in this nice webinar. This is the first problem, and the other problem, of course, that I want to address and was already addressed somehow by the previous speakers from India, is that the clinical decision systems and the international clinical guidelines barely is adapting, maybe adapted to our grounds. As you know, in India happens, Abdul, you know well this. Usually, you know that the international clinical guidelines, IDSAs, for example, more than European IDSAs, are not thinking in low and middle income country contexts. And usually they will give you some recommendations that with drugs that are not already available in our pipelines, that are not ready available in the countries, or even if they are available in the countries, they are too costly, as ceftazidimab, Bactam, or Astronam, that are not usually present in the hospitals, and that cheaper alternatives. We'll talk further probably about gram-negative multi-drug resistance. For example, ceftazidimab, Bactam plus Astronam is a nice combination, of course, but we usually use also colistin, tigecycline, fosfomycin, etc., or aminoglycosides or meropenem according to the MIC. And usually in the clinical guidelines and the clinical support systems, these alternatives are not contemplated. So we might have first available terminals for prescribers in surgical unit, in ICU units. Now in my hospital we have them, but not interconnected with cell labs. I have more ideas for later. It's not to speak too much now. Thank you.
Right, yes, we are all interested in AI, but none of us are AI experts, and we specifically did not invite a real expert because we are not really interested in the technical details. We want to discuss, we really need it, and if need, what we are expecting from the real experts. Nitin, we both work in oncology centers, a lot of febrile neutropenic sepsis. We are starting a patient with neutropenic sepsis, shock, meropenem plus colistin plus teicoplanin plus caspofungin. After 3 days, we want to step down. Will this AI users help us to step down? Is it possible?
I think, sir, definitely. This is what I was trying to convey to the initial in your initial question when you asked, do we actually need AI? So when I was training under you, sir, I think in 2016, 17, and 16 to 18, when we used to conduct how I learned to do antibiotic stewardship was there was a list of high-end antibiotics which was given to us, and the pharmacy would give us the list of patients who would be receiving high-end antibiotics, and then the pharmacist or the ID physician is supposed to review those patients whether they're actually required or not. So out of my curiosity, or now I can say out of my ignorance, I would have asked you someday that why are we concentrating only on high-end antibiotics? The automatic answer from you came out to be, I cannot see each and every patient who is on any sort of antibiotics. Like I cannot see a patient who is on cephalosporin sulbactam or who is on comaxiclav or amikacin. That the volume is so huge that we cannot see. So by the time we see the patient, like you and I see the patient, we are already losing the battle. Patient is already having a carbapenem resistant bug, or even sometimes even VRE or a candida is already growing. So in order to catch the bugs earlier, or in order to outsmart the bugs, we have to catch them early. So we have to see each and every patients who are on even first line antibiotics also. So for that we need I work in a 500-bed hospital. Almost 250 patients are on some or the other antibiotics at any point of time. It is it is inhuman for me to see every patient every day. For that I need AI. So in order to save antibiotics, I think that's where the volume part of it, particularly in India and in high volume centers, that is where the data management team, data sciences, and the like all the software are going to work. They're going to basically like prompt the clinician that your first line antibiotic itself is not needed. You don't need even an ID consult for this. You can stop those antibiotics. I think that is where the real impact of the softwares will come.
Yes, Nitin rightly said. We were overwhelmed with number of people on antibiotics, including high-end antibiotics. We need AI assistant for inpatient antibiotic stewardship. Vasant, what do you think? Do we need AI for outpatient antibiotic usage? We don't prescribe meropenem or colistin, OP, but still, is AI going to be helpful to us in outpatient antibiotic stewardship?
I think the where I'm working is digital, completely digital, 100% digitalized hospital, and I think this AI, if we implement into this in the digital data, will help us to get the pattern of resistance and also help through get through the genomic analysis, and definitely it will guide us which antibiotic should be prescribed on outpatient basis, because whatever data will be generated from the microbiology lab and the prescriptions audit which happen will guide what should be the pattern of prescription at outpatient hospital basis and what should be the prescriptions in the in hospital inpatients. So definitely I think AI would, because many of the doctors in India will not follow the antibiotic policies or something, but I think once a clinician feeds in the data, possibly the AI comes in play and will help the clinician to prescribe what would be the correct choice for that patient.
Thank you, Vasant. And so we need AI in IP, we need AI in OP. What about microbiology lab? We need opinion from Raksha and Yamuna. Raksha, do you use AI in microbiology lab? Do you really need it?
I would say at this point of time, partially yes, sir. Because when you say clinical microbiology, you deal with a lot of data in terms of images or say graphs, curves, or data points which you're collecting, antibiotic susceptibility patterns, antibiograms. So when I have such high volume of data, and as Dr. Yamuna mentioned, that we have lack of expertise also in our country with reference to clinical microbiologists per se. So when you look at the manpower, we definitely need AI to help us in clinical microbiology. But when it comes to implementation, how much of your lab is equipped with these high-end automated tools is the question. Many of the labs still follow manual processing of AST. The automated systems are not available. But if they are available, of course, you are using AI to a large extent there in terms of plate reading or interpreting the AST patterns or entering that data into your hospital information systems, getting antibiogram or using a simple WhoNet. Everywhere you have a component of AI in clinical microbiology. It is not that it is not there. It is partially there. It all depends on how well your clinical microbiology department in the laboratory is trained to use this AI as a tool and give that data to your clinician to interpret the antibiotic sensitivity report that you give. And it should not be just a report. It should give an interpretation and what to do with the report. I think AI helps clinical microbiology there.
Right. Microbiologists are also asking for AI. Now, what about infection control team? Yamuna, do you really need AI for infection control?
Yeah, I would just like to bring in the example of radiology department, which was the first one to go with AI. And what they saw was the X-ray reading, which is like an enormous level of data, which fills in the clinical skill of the radiologist, whereas the time given for them for the actual CT scan or the MRI, where they have to spend their time on a larger time basis, was getting affected. So when AI platform was integrated with the different types of X-rays, and when it was given into a deep learning system, the part of the work which was attributed by the radiologist only to visualize an X-ray, not only coming in their hospital, but also as a telemedicine or also from the outpatient department, when the workload gone down, they were spending more time on the critical decision-making areas, which are the CT scan or the MRIs for the patient. So same way, 50% of the time when IPC comes in, the workload goes only doing surveillance activities. Whereas the only 30 to 40% are the time when people are available at the bedside of the patient. So if this surveillance effectively is taken care by the AI and we can integrate this data for the predictive analysis, we will be doing more work on the floor for training and giving feedback for the people. So let me just quote few examples recently published in Journal of Hospital Infection, where they incorporated a Sure Wash system, which is a commercially available interactive kiosk, which was a camera-based augmented reality, which was a gamified learning platform to train, assess hand hygiene, and also give a feedback to them. And they integrated the audit mechanism. And they saw that the compliance and the quality of hand wash or the hand hygiene improvised in their hospital. And also one more study published in Egypt in 2020, where they incorporated the computer vision-based AI platform in the surgical ward. And when they saw that the hand hygiene compliance by the surgeons coming to the ward improved by more than 90% by their quality and quantity. But again, it has to be completely integrated with the feedback mechanism. The moment the feedback mechanism came down after auditing, cognitive offloading also happened. So people forget if automated feedback mechanism is pulled out. So it is a plug in and plug out. If it's plug in all the time, people cognitive level on compliance would also improvise. Not only that, there are certain smart IPC system which can be incorporated in tertiary care, which was published in the International Journal of Medicine, where they saw the important IPC practices, which takes huge time of the practitioners or the IPC practitioners in doing auditing, is a personal protective equipment adherence in isolation areas, or let it be in OR, how well the people wear the PP, how well the PPs are guided or taken care inside a critical care area. If we have an AI platform and a feedback mechanism, there is definitely more than 15% reduction in hospital acquired infection. And if we could integrate it with certain AIs, with Dr. Raksha was mentioning, like the forest model or the convolutional neural network, we can also look into the predictive analysis. If my PPE compliance in the OR is not good, if my OR traffic regulatory mechanism is not good based on the sensor or the movement sensor, I can predict that this patient can end up having a surgical site infection. And it can contribute as an alert mechanism in reducing 25% of this predicted surgical site infection. And these are very important published data which has already been looked into, and thereby we can improve the WHO guidelines by the adherence platform, which has to be done to prevent surgical site infection by more than 85%. So these AI platforms can definitely help us to do a real-time monitoring, to send feedback, to predict what can happen to our patient, and thereby do a real-time corrective mechanism and reduce the hospital-acquired infection. I think AI is definitely a way forward for us to reduce the unnecessary surveillance, which is done only in the morning shift. We hardly do any surveillance activities in the late evening or in the night shift, where we do not know what is happening to our patients. So these things would definitely help us to improvise our practice, predict what is going to happen to our patient, and thereby bring corrective mechanism.
Yeah, Dr. Yamuna, we all agree, AI is necessity in infection control. It will bring out the reality, what's happening behind the screen, what we usually sweep under the carpet. AI will bring out everything. A bit dangerous too. And also there's a concern. One of the senior radiologists, my friend, told me, Gafu, AI is very good, but we radiologists are scared. Some of us may lose our job. Will the hospital think, now we have got AI, we don't need these doctors, why should we pay them? Is it a real concern? Pierre and Euron, what do you think? Are we going to lose our job?
I just wanted to react on this great story by Yamuna. I think it's really, really important, but the main thing that I think I hear you say, it's not only registering what's going on, like you can see those movements. You put a camera somewhere, you can actually calculate what the movements are. It's also the feedback, right? What are you gonna do with the data? And I still feel that there's still a big gap between actually controlling what's happening, ascertaining what is the gap between what should be done and what is actually done, and then influencing the people, right? So there's the other thing. That's a human condition. Still, changing your behavior is a very, very difficult to change part. That's not something that comes freely with AI, right? It still would ask you to think about behavior change techniques. And I think what is really interesting, and I'm very interested in that part of the game, is that how are you gonna ascertain which type of barriers, which type of reasons why people are not adhering to a certain behavior? That can be prescribing behavior, it can also be IPC, right? How does that come? And I think that there's another place there where we know there's a huge amount of reasons why people don't abide to the guidelines, and it's very different from place to place. It's very different from objective to objective, right? If we look at starting antibiotic, it's completely different from stopping antibiotics in terms of the type of behavior you have. So I'd be very interested to see if AI could help us to analyze that kind of data, which is very contextualized again, and help us to find what would then be the best method to improve, because feedback is one, but feedback doesn't always work, right? You still need to think about other ways to make that work. And I think there's a big opportunity also to see what type of behavior would work in what specific circumstance. The surgeon is a different person than the hematologist, than the ICU physician. The circumstances are different in every different hospital. So thank you very much for that really great incentive on using it in IPC. But let's not forget the intervention part of it.
Yeah, yes, Your Honor. Agree. Raksha, we all know that ChatGPT has the habit of hallucinations. I tried papers, it give you references that do not exist. Once I did my own article search, it gave me a list of articles I've never written, I never published, and the typical hallucination. And what can we do to prevent hallucination in antibody stewardship?
Again, when you're talking about ChatGPT, you're looking at a large language model. And you must understand that the data that you're feeding in there, it is already analyzing based on the existing knowledge that it has. So there are definitely gray areas here because multiple people are feeding into it and there is no standardization and no calibration of this data. So a quality control system of these kind of LLMs is something which we haven't worked on. I mean, I have not found any recent literature or studies that which look at concepts like model drifting, where you have already trained a model on an existing data, but of course there are things happening. New patients are coming in, new ID concepts are coming in. Now, what is happening to LLM when these new concepts are coming? Are you retraining your large language model again based on that data? So these kind of questions do come into picture. So how do you train the large language model or a machine learning system which you have made for your setting based on the continued data that you're generating in your setting? So this recalibration concepts of looking into model drifting and looking at the feedback, what you're giving to the model, is it going to
Raksha, can you explain to us what is this drifting, model drifting?
So what happens is, say, for example, we are currently in 2025. I'll try to simplify this. ChatGPT came when it came initially. I think we were using the 2.5 version. Now, if you go and see, it is 5.1. So every 1 month, a new version comes in. So you can try it on your own. If you are going to type in a question today, like say, what are the articles written by Dr. Abdul Ghaffur today? And after 2 days, you type the same question. It probably might give you 4 more articles which you wouldn't have written. So what's going to happen? It is going to look at the question that I have fed in from, say, Bangalore, and what you feed in from Chennai. So 4-5 questions you feed in, it's going to continuously learn by itself. It might be right or wrong. So it's going to drift over a period of time, and you don't have a standardized database there. So data analytics is a very gray area for experts like us in other fields. So I think we need to work with these systems in line with the data analytics to see what kind of prompts are we building, because everybody is feeding in questions, and the questions that you feed, it is going to train, retrain, learn, unlearn, relearn, and it is going to be a Pandora's box if the right people don't feed in the right data. So I feel we have a lot to do here. Even if you want to make a model for your own self, it is not a one-time model. You have to keep it in your pocket, like your mobile phone, and it needs to be like the newer version every day keeps coming, and you have to update it every time. So that model drift is all
That
under
means the bugs become smarter, bugs are changing, antibiogram is changing, patient response is changing. We have to update the model. A model
Yes,
developed a year ago, we
it
can't
has
just
to
use
be
the same
real
model
time.
today.
Yeah,
To retrain it. That means the AI needs some continuous
continued
medical education or
inputs.
AI
Yeah,
education.
yes.
Education. Now, next question, Nitin.
So
Yeah,
Dr.
we start implementing AI. One
Dr.
specific
Daniel,
question.
So Dr. Gabby,
Yes.
Dr. Pierre wanted to have.
Oh, Gabby. Yes, Gabby.
Just the other side of the moon. My problem with that resignation, I don't understand anything about machine learning. I know something, but I'm not an expert. Is that what is happening with the prescriber? My great issue with AI, with the ChatGPT and all the usual, the common tools, is exactly that's what I raised my hand. Exactly what you say: the hallucination, the reference that don't exist, or reference of bad reference or reference. Usually, if you make a look, usually for writing something in stewardship, the references are websites more than articles, and articles are of not quality. So we are always have to be there to teach and to be alert when young prescribers, UCI, etc., gave me back. Gabby, AI said me that I have to treat that coagulase negative staphylococcus bacteremia. Why? Yeah, 5 days, 7 days, wherever you go, because it's in a guideline. What is the level of evidence of that? That is awful. That is the problem. We can rely on this. We can simulate to this, but critically analyze that. And that's where our brains, our old brains there to try to face this issue, because if not, the harm will be much more important than the benefit.
Yeah, what Gabriel mentioned, if the AI model give us an answer, a recommendation, it should substantiate and back up with guideline evidence, not some vague ideas and maybe hallucinations. Pierre?
Yeah, I just wanted to react when you asked a few minutes ago, are we afraid that the AI will take our job? And I think it's interesting question. I'm not afraid of it because I don't know about any country in the world where we have enough healthcare workers. We have staff shortage in every hospital in the world, probably. So I see more AI as a tool to save time for us, to spend less time trying to analyze data on the computer, and to spend more time with the patients to explain them what we would have seen, what we would have learned from the AI. But we have to remain the boss, and I totally agree with Gabriel about just what he said.
That's a good point, Pierre. Radiologists may be afraid of losing job, but we not antibody stewardship team or infection control expert. We are short-staffed. We need AI expert assistants. And Nitin, we are using an AI tool. After 3 days for antibiotic step down, we're using the recommendation of AI. And it give us a wrong advice. We are following it. Who is responsible? We, the hospital, or the people who develop the tool?
Sir, again, there are two aspects. One is ethical aspect, one is legal aspects. Legal aspects wise, I think whatever I know, I'm obviously not a medical legal expert, but I think as far as today's concerned is goes, whatever AI tool or whatever tool you use, it is a person's responsibility that the decision made out of the tool is right or wrong. So from that point of view, we are responsible. If I am using that tool, I am responsible. That is the legal aspect of it. Ethical aspect of it, I think, will evolve over time. Once we start using it, then only we will know that whether ethically we are right or not, because there will be various aspects of using various machine learning tools in future or currently also if you are using. So as far as, I mean, short answer is the person who is using, the person who is signing off the drug, or whether he is discontinuing the drug, is the whole and sole responsible person. We cannot push the responsibility to some other person, either to the hospital also.
Yes. Thanks. Next, Yamuna.
Sir, yeah, of course, as I agree with Dr. Nitin, what he mentioned. As far as ethical issue goes, it is still with the clinician, because the AI is not a rule-based. What we look into is the machine-based learning, and machine-based learning comes with the practices which has been happening over a period of time, and we need to keep incorporating the expert opinion on all that. So machine learning is not something which can be driven on its own. So every aspect of machine learning has to be incorporated with expert opinion, where they call it as certain models which are staggered model, where every aspect they would require a smart human innovative data which has to be fed in every time in order to make a usable decision. So those models has to be adapted in place so that we don't mistake or make such decisions which would end up later in an ethical issue.
Right. So as Yamuna mentioned and Nitin mentioned, yes, the prescriber is responsible, but it's a hospital who is purchasing an AI tool from a company. So hospital has got a responsibility to purchase the right model for the right price, not some low-cost model. And developers also have responsibility. They should be transparent about the model, about the training, about the validation. So everybody has responsibility, but the final responsibility is with the prescriber. Dr. Vasant, what are your concerns about you using an AI
I
model?
have just one statement to make is that AI, if the clinicians, AI cannot replace clinicians, but if the clinicians don't embrace the AI will replace the clinicians. So I think we'll have to keep ourselves updated along with the AI. I'm not saying that AI is going to be the final authority or something, but we have to be updated and AI cannot replace clinicians, but again, the same statement, if the clinicians don't embrace AI, the AI will definitely replace the clinician. So we have to be
Rightly said. So AI is not going to take over our job, but if we don't learn AI, another infectious disease AMSP consultant will take our job. Euron?
Yeah, just one aspect that I just came across that I was thinking about is that so because we were talking about uncertainty of decisions, right? So sometimes if you ask ChatGPT or whatever other LLM, you ask him something, and then they will say, this is true, or this, sort of the level of being sure about something, right? And everything we do on a day-to-day basis, there's always a level of uncertainty in it, right? There's never a guideline that has a recommendation that is so 100%. Well, there are maybe some recommendations, but most of them are just fraught with uncertainty. And I think that's a really important aspect when you judge the value of an LLM, or that this machine recognizes that there is an uncertainty here, and that uncertainty will always exist, right? And that's nothing to do with the literature or with the evidence. It's just for sure that nothing is for sure. Nothing is 100% for sure. I think that's a thing that we're still missing in many LLMs. When you type in something, you get this answer, like it's like clear, like whatever it is, it's a very clear advice. And I think that's where we need to be very careful. There is an uncertainty in everything that we do.
Thanks, Euron. Now, we know that we need AI tool. We know it should be affordable. We also know that an AI tool trained in US or UK cannot be immediately used in India or Argentina. We need local validation, local machine learning, and training. Now, APUA being an international organization, how can APUA contribute to usage of AI, introduction of AI validation in various countries? Pierre, over to you.
As I am not an AI expert, I am probably not the best to address that, but just want to thank you again for organizing this webinar, because I think it's the first step to try to get APUA more involved in the process. I'm convinced it's important, and I think we need a meeting like we are doing today to exchange about this, and that everybody in the world tries to think about this.
Thank you, Pierre. The right way forward. Euron?
Yeah, well, I think one of the things that I would really find interesting is that in any part that where we work in, there are tools, right? So people develop tools all the time, and I think that having APUA being able to provide or to collect tools that are out there that then obviously would need contextualization would be valuable. Right. So and that could be a start. Of course, APUA is not an organization that can easily, either fund or organize large studies on AI, but having collecting valuable tools from different environments and making them available or drawing the attention to them for our members would be valuable, I guess.
Good. Thanks, Sharon. Gabriel?
Yeah, to complement what Pierre and Sharon had said, first, I was looking when you made the question about the foundation, the funding of this. We cannot work alone, of course. We are not a funding organization. We are far from that. And so we should look for stakeholders that can put the money first to have the computers, the terminals for prescribers. This is the structural part of the component of the stewardship program, the structural, the computers, the knowledge, and the interchange, as they say, some of the people said, with the micro lab and pharmacies, that we can use really at least the minimum for AI for alerting the blood cultures with the blue carbon positive or film array if you have, or whatever you have in this patient, alerting quickly, then sending the recommendations about which are our sensitive or resistant patterns. This could be the minimum we should try to do. That from the structural part. And from the other part, of course, trying to evangelize about the things that AI is not all if we don't have enough smartness, the prescriptor, to understand that that recommendation should be taken with very conscious, with very, very carefully, because if not, the harm would be most higher than the benefits. This is important. From the structural part, we can try to look for founders that can help us to implement customized AI. We should be taking care about what these programs are working and what are recommending, and we should be, there are three parts: the teaching part about the understanding and properly use of this. Thank you.
Thanks, Gabriel. Now, see, our theme of the webinar is crazy ideas. Now, let's ask a few crazy questions about AI in AMSP. What is the craziest? Now, first, Raksha, what is the craziest AI idea in stewardship that you genuinely believe India should adopt in the next 5 years? You can be craziest person in India.
When I started thinking on using AI, I always believed that AI for doctors is one thing. I feel AI to a certain, I don't know how crazy you would call it, probably for patients also something on AMSP at a community level, if you could develop something to educate them and make them, okay, there's an antibiotic prescription there or a strip there, it should give them an alert saying, no, this is danger or something, no, you're not supposed to take it without a prescription. Something like that. I want to make it personally for the general public or patients, where antibiotic awareness is there in the community and AI goes there. I mean, that's my current craziest idea, what I can think.
Thank you, Raksha. You are a very sensible person with a lot of common sense. Crazy, but we need craziest idea.
Currently, that's what I'm thinking.
You're a very practical person. Nitin, you're a bit crazier. We need the craziest idea.
So, I mean,
to inspire you before that answer, Steve Jobs once said, those who can change the world are the ones who change the world. And you want to change the AMSP scenario in the country. So your idea should be very, very crazy.
So, I think one of the important aspect of AMSP is interpersonal skills, sir. I think how you articulate yourself and how you put forward your idea of communicate your idea to the prescribing physician, surgeon, hematologist, oncologist, or whatever, it matters a lot. So I am not a very articulative person. Neither do I dress well, and neither do I smell well. So sometimes my ideas are not taken in a very good way. So that's where I feel it's sometimes a non-personal, like non-human tool as a much more role, because that non-human tool can dress, smell, and everything can be done in an artificial way to sort of seduce that person who's prescribing that antibiotic, that yes, prescribing that antibiotic is not right. So I think that is where we should aim.
Yeah, Nitin wants a very handsome or beautiful AI robot walking in the hospitals, knowing everything, controlling all antibiotic stewardship, controlling all the prescriptions. An excellent idea. Yamuna?
Yeah, I'm not getting any crazy ideas. Very difficult to think. So. The only crazy idea right now getting is to get an AR-based glass so that when I wear, I can really see people who are not the appropriate performers, and I could send them the real-time feedback then and there that they are not performing or not compliant with the practices. That's the craziest idea, so that I can get it and give it to everybody so that they can look into it.
So you want an AI specs immediately live will give you a live idea and hygiene steps. Send to a computer. Amazing idea. What's the best idea?
So I think over-the-counter prescription by some way, if they can be stopped completely, which is a major concern for antimicrobial resistance in India. Somewhere, AI comes into picture and prevents a complete lock on those people. I think that is what will help a lot. And also AI at primary and secondary care centers, much more than the tertiary care centers. If we have something of that sort, is I think locking completely these people over the counter prescription, which doesn't happen anywhere. I think that is very important to stop that.
Thanks, Vasan. Puron, what is your craziest idea?
Yeah, so I can't judge for India, but I think that what my craziest idea would be is that as an AMS steward, I would be doing my handshake stewardship at the ward, and I would be analyzing the discussions that I have with surgeons or ICU physicians or whoever, and that by analyzing those conversations and looking at determinants of behavior, right? So looking really at incentives that that would work for that particular group to change their behavior, having that analyzed and getting fed back to me immediately, so I know which kind of approach I should have to change prescribing behavior. But that's sort of my instant AI dream, but I'm not sure if that's gonna ever work.
Yeah, good, because that's our good crazy people, intelligent crazy people with crazy ideas. Now we need a French craziness, Pierre.
I think my best choice would be to try, because we know that most of the problems we have with AMR and with antimicrobial stewardship is that there are some prescribers that they don't listen, they don't want to know more, they just want to do what they want to do. And I think if the AI would allow us to identify who are these outliers and how we could convince them to go back in the reality and to understand how bad it is that they don't care about what they prescribe in terms of antibiotics. So something like an individualized monitoring or screening of bad behaviors between the prescribers, and then we would have, as human beings, to go to them and to try to convince them to go back in the good way.
Yes, yes. Gabriel, now crazy ideas from Argentina.
Yeah, but again, my crazy idea for Argentina, for Latin America, as I said, regarding that governments, the commitment of authorities when we work in America Latina, regarding the support of the hospital and antimicrobial stewardship, should be my crazy idea is first AI that convinced authorities that this is the right way and invest in AI, in artificial intelligence, so we can have artificial intelligence in our hospitals. And then, like said the women and Pierre, that apart from the alerts with the proper recommendations according our sensitivity and our pharmacy stock for treating bad bags, that the same every day sends a message, for example, to these prescribers or the bad prescribers, as Pierre mentioned. Remember, are you sure you are doing well? It's okay, you're patient. Do you need some help? Something like the human that is collaborating. That would be crazy. Of course, I don't know how to do it, but probably would be useful, more useful in Latin America. Systems and some feedback, some crazy feedback. Please don't forget about that. Please remember that I was advising you with the proper treatment. I think that is crazy, probably.
Good. Thank you. Thank you. All crazy ideas, but workable crazy ideas. It's 9:30 p.m. in India. We are half an hour behind beyond the schedule. Any more interesting, important points that panelists would like to speak? Any one of you, anything really crazy that, if you don't express, that will be a big loss to the world.
I just want to express one word into it. So whenever we are doing any data analysis, we need to have the social factors also added into it. The ethical practices also added into it. Ethnicity also added into it, because the data which is common in Italy, data which is common in Europe, we cannot adapt it and bring it to India. For example, if I have more of patients getting admitted in southern part of Chennai, I will have data only from patients admitted from southern part of Chennai. And I would be thinking that these are the patients who are having high level antibiotic exposure or developing antimicrobial resistance. Or if I have more of international patients coming only from Africa, my data will be completely filled with patients coming from Africa, where I would think only the African patients are getting more of antimicrobial resistance as compared to other patients. So we need to look into the social factors, and the global factors has to be incorporated. Even when we talk about IPC, social factors has to play a very important role, so that it also gets integrated into the AI platform in order to make the diagnosis the most important, and globally it's acceptable worldwide. So that's what I want to integrate, not only the clinical data, but we need to fill into every time the social and ethical data into when we are looking into the AI platform or moving way forward into the active learning platform.
Yeah.
Thank you, Dr.
Also,
Yamuna.
sorry. Also, probably to add with
we
that,
have to, come to an end this warm, amazing webinar. Thanks to, yes, Pierre. Gabriel?
Yeah. I was trying to add also to what a doctor said, that these messages can add to that alerts to the prescribers. Do you know how much you are spending today with that bad treatment? To try to send messages of awareness, of cost, of microbiota harm, etc. That could be also the strawberry of the in the dessert of these messages, these crazy messages that can bring the prescriber some guilt about what they are doing bad, because now we are all here because someone, a lot of people is doing bad regarding prescriber antimicrobial.
Right, Gabriel, thank you. Any other comments?
One more crazy idea. I think just those clinicians who do not follow the proper antibiotic policy, using, haphazardly using antibiotics irrationally, especially in tertiary care or secondary care centers, they should be banned from that coming to the hospital for a week time in beginning, and maybe punishment for a month's time future. Then it will get them proper dose. Similarly, for surgical site prophylaxis, if the surgeons don't use that surgical site prophylaxis according to the protocol, they should be banned from operating for a week's time. So I think automatically everything will fall in place.
Wonderful. That's I want.
It's a crazy idea, and we can't ban the surgeons from the hospital. That's not going to happen, but it's a crazy idea. Good. Thank you all the speakers and panelists, participants. The crazy idea shaped the AMR world we stand on today, and we need more such crazier and craziest ideas to sustain the momentum in the world of AMR. Thank you, Apua. And it's a joint meeting by AMR Declaration Trust and Apua. an amazing meeting, and we should take more such meetings and ideas and events where we can work collaboratively to tackle the global challenge of antimicrobial resistance. Thank you, attendees from across the world. We have got attendees from dozens of the countries attending this meeting, and thanks for making this webinar a great success. Thank you all. Good night.
Good night.
Thank you. Good
Thank you.
Good weekend.
Thank you.
Bye bye.
Thank you, sir. Thank you.
Bye.
Thank you.
Bye. Thank you.
You.